inovação estão sendo a menina dos olhos do evento. A gente está super orgulhoso do trabalho de todo mundo. Eu acho que só quem está aqui na frente do trabalho, todo mundo que está aí assistindo, participando da gente, também trabalha junto. E agora a gente gostaria de começar o dia com a apresentação do Daniel, da Biosim, e ele vai fazer coisas bem interessantes para a gente, para que todo mundo aproveite bastante a apresentação do Daniel.
Good morning, everybody. So today we have the pleasure to be here with you. I'm from the Inovation Committee to invite us to this important symposium. And the idea we have today is to introduce you all the possibilities that Biosim has for the English part. So we are a company that is based in India. And I'm personally take care of all the American market and all the process implementation and development of the American market. So the idea is to show you all the opportunities we have in this years. And at the end, the presentation will have a video. The catalog that we have, so this is general idea. We have some we will cover 4 main points. General introduction, to introduce a safer vaccine to prevent the disease. There is also a political government monitoring all this process, allowing you to get a better characterization of the process and the product you will have. And also, a lot of people is moving to the fully automatic facility, where you can diminish the number of the people involved in this operation, etc. And Biosim is focused in these 3 main objectives. We are not a company that are focused in disposable. In the last conference yesterday, GE, of course, they are focusing disposable. We are not focused in disposable technology. We are focused in stainless steel technology Actually, also we can do hybrid system when you can have some operation in stainless steel and you can join this in your process flow diagram with the disposable application. But we are not a company focused in disposable. So here, as I thought, we have process technology, we have inline monitoring, real-time release, and we have fully automatic facility. So all of this we try to integrate in a package that we will see now. Sorry. So here, if you see the pharmaceutical products cycle line, you can see also that, of course, disposable is very well used in research grade and even in the first stage of the process when you have phase 1 and phase 2. But sometimes when you need a very big scale, even above 2,000 liters of fermenter of bioreactor, it's very difficult to move to disposable. And also it's published that never stainless steel will be fully removed. So there is a big discussion of the advantages of disadvantages to use and not use bags. And of course there are several advantages, but also stainless steel remain to be a technology that a lot of people continue using. More when you are trying to do a very big scale of production. So based on that, I will show you some slides when you can see some pictures of the equipment we are developing. This is our, let's see, our stimulus. We are in the process of saving life because, of course, the equipment we are developing is involved in saving life in such a way. And we have mainly three main activities. We have the design build, so everything is designed in India, of course, with all the suppliers from Europe, US, etc., like stainless steel shelf, all the material, electrode, etc., are the reputed material. We have also a training, research, and regulatory service when we train the people in bioprocess. And after that, we have the integration service where also we are all related with the filter validation, etc. It's good to say that this company grew from Sartorius. So this company was created in 2004, around 2004, and was created from Sartorius, India. So the owner of Sartorius, India, separate from Sartorius and create this company. So all the knowledge related with filter, cross-flow filtration, etc., we have it in the company. So we have two main areas for production where you can see there. One new area is also inaugurated the last April. So, and in this facility we have more or less one capacity of produce around 4 bioreactor of 4,000 liters per month, more or less, of stainless steel, depending of the complexity. Here you have some pictures of the facility. This is a new facility near to the airport that was built for increase the capacity of production. We are very focused in we have product of course in Europe, but we are very focused in the development vaccine manufacturing network. So a lot of people from India, Iran, Cuba, now Brazil, we have starting projects with Brazil, and hope we can continue. So a lot of, in India is the country where you will have the biggest mass of production in terms of volume in biological in the world, even more than Europe in terms of volume, I mean. So India is a very big market also for biological. And we have also in India the Serum Institute of India, that is, is one of the biggest producers of vaccine over the world. So our model of development, we are, as explained in PET mold. This is a mold that we have process, equipment, and people. So we pass for the design of the solution where we can design bioreactor, fermenter, CIP station, filtration system, biokill vessel. You will see later some example. We have a full automation department when we can do all the automation of the facility, all the automation of the equipment and integrate. We have also the integrated service when we can also be involved in the facility layout, etc.
We have also a research unit when we train the people. So a lot of people that come from the university before going to the industry go there and be training. Also people from the industry train there. The training can be in process, in validation, in engineering, etc. This is the manpower training. And we have the regularity services, that is when you make the validation of the filter, etc. So as explained, we are focused mainly in those equipment. So media vessel, buffer vessel, conjugation vessel for any conjugation vaccine, reactor vessel in general, fermenter and bioreactor, any volume from 1 liter to 15,000, 15,000 liters, so 15 cubic meter. Cleaning in place solution, station, kill vessel, and also we can integrate all in the facility. So if you see this slide, here in green is everything we can cover. As explained, we don't have a fixed catalog, everything is customized. So you have one need, you have one space, you have one budget. So we are flexible to customize your URS according with your requirement. We are not manufacturing chromatography system, and also we are not involved in any relating with filling and packaging. So these three operations, we are not involved. But all related with the filtration, viral filtration for removal of in mammalian for remove viral, cross-flow filtration, etc. We are involved. So some of the main clients. So here I would like to show you some pictures. So here you can see, so it's a real picture. So here you can see some fermenter for acellular pertussis production, one for anthrax, this is for Korea, this is for India. Here you can find continuous 3,000 liter perfusion system for mammalian cell. So very big fermenter. When you have 6 perfusion device of ATF for the production of MAP. Here, one of the publication of biosyn in the microcardial culture for virus. Here some super skid. So we are talking that some of the application we have is a very big scale. We are talking about a skid of 6,000 liter each vessel. Yo, you don't, oh, sorry. But you understand everything or no? Sorry. Okay, for the translation. Okay, sorry. And okay, this is the super skid system, where you can have several integrated vessels that you can use also for buffer preparation or for media preparation. So this is also another super skid view from the top. Here you have media preparation vessel, some examples. So even you can see different possibilities. You have a small space, we can design a smaller station. You have, you need one level of automation, another level of automation, depending on the application. So everything is customized. So you have one integrating skid for combined vaccine. So sometimes this is very easy. You have a pentavalent vaccine or hexavalent vaccine. We develop equipment for this type of application. Some small vessel if you want to, if you need some small media vessel or buffer. If you need cleaning in place station. So if you need to develop a central CIP station or mobile CSP center, or if you want to clean all the vessel, or you want to clean 3 vessels, or any of the possibility, according with the URS, we can manufacture. So you can see some example of the CIP. So kill vessel, as you know, a lot of the facility that involve pathogenic microorganisms need to inactivate the waste, so need a waste treatment. So also we have the application to the inactivation system in 2 versions, in the continuous one and in batch. Here you can see 2 examples. So also, as explained, you have cross-flow filtration. In the, even here, you can have in the hollow fiber mode or in the membrane plant mode. Also, we developed the holder. Of course, the filter belongs to Sartorius or PAL or Millipore, depending on the or your need. So some small cross-flow system. You can see here, some small. So you see that everything is not so fixed. So depending on the, in this case, depending on the membrane area you want, depending of the volume of the you want to process, etc. So some example of the cross-flow system. So later, when we have this all several equipment and you want to integrate, sometimes the integration is very important. Sometimes not always is very important because you need to clean the interconnection piping, you need to do CIP, you need to do SIP, etc. So you need to integrate all the system. So we do the hand check and the communication between all the system to allow you to clean and do the completely integration of the facility. So like you can see here. And even if you have some equipment that don't belong to BioSync, also we allow to integrate. Suppose you have a centrifuge that we don't produce, for instance, a Westphalia or the Sharper centrifuge, or you have a GE chromatography system, also we can integrate. Also the automation that has explained you is one of the main objectives. So we can integrate in a SCADA different suite. You have one suite for downstream, you have one for upstream, or you have several suites for downstream, so we can integrate everything in the SCADA. Here, for instance, you want to integrate several fermenter or bioreactor with the media preparation or the buffer preparation. Also, we integrate all this in the SCADA, and we guarantee all the cleaning between the transfer piping between them. So something like this. You can see the full integration. As explained, you can integrate one unit. You have one unit, or suppose you have one cross-flow or you have one fermented, you can integrate. You can integrate several units or several skids in the same suite. Also, you can integrate the process with the CIP and all the interconnection piping. And if you have a building managed system, also you can integrate the equipment with the BMS system. So there are some examples when you have a single system, PLC and HMI. I will not go in too much detail about this. Multiple system application, common system, when you have a redundant service or you have a redundant PLC also. So all the kind of, depending of what you want and the money that you have also. So here, this is a publication of Siemens talking about the potentiality of BioSync. So. And now I will go to some particular design for some application, just only for show you some of the application we already make and the result in the production. So here is very important. We follow this strategy. So you have one product, you have a critical quality attribute, you are trying to get it. You have your main critical process parameter, and we design the equipment based on that. So we have experienced people in process in the company, and based on the quality you are searching and what you want to monitoring, we design the equipment. So we follow this, so you will not receive a standard equipment, you will receive a fully customized equipment for your application, regarding with the quality attribute you want to control and the process parameter you want to control. This means that all the recipe we made in the automation program is a recipe for your application. It's not a general recipe of automation. It's for application. If you need, suppose, any chemical inactivation or transfer, or you want to control in such a way the fermenter or the bioreactor, we develop the sequence for this application. So there is not anything is a standard here. So we pass for the technical complexity. When you have a critical process parameter, we have the GMP requirement, and we have also the risk management. Also, when we take in consideration the risk management, also we also take in account the training of the people. So we train the people in during the pre-FAT, during the FAT, and during the SAT in the site. And all the process know-how that will help you to the design of the equipment. So this is an example of process flow diagram for MAP. So you can see a lot of process parameter and quality attribute that you need to follow for having a good quality product. And this we are taking into account when we design the equipment. So here's some of the sample of bacterial product. Here you have, for instance, tetanus. This is another auction we have. You know, some of these vaccines are very old, but the life continues. So people want also to upgrade what they developed 20 years back. So we are introducing new sensor, new way to do it, to update the facility because tetanus you need to continue producing, because you need it for DPT, for Penta, you need for conjugation vaccine, etc. So also we developing new process and new design for achieve a very good yield of tetanus toxin in this case. So you can see some P&D diagram of tetanus dedicated of this. So one real picture. This is also producing since the last 15 years with a very good yield of tetanus. Here you have a cellular pertussis product. In this cellular pertussis vaccine that is also used in the pentavalent or hexavalent vaccine, you have 3 main antigens that you need to promote the expression in the supernatant or in this process. So we developing a completely process for this, having in consideration the same we discussed, having the consideration the critical process parameter and the design of the equipment to achieve this process. So some of the sample of cellular pertussis P&D. So here you can see the real picture in the facility where they are producing. So this is in this case one 3,000 liter fermenter and one 50 liter inoculum fermenters. So one for dystheria here you have also very important. So the same, one designed specifically for this when they have a very good yield in the last 10 years. So very important pneumococcal production. Here you can see one very big skip for pneumococcal production that Biosyn also implement. Here the P&D, well, all the characteristics for the growth of pneumococcus, specifically like you can see here. So glucose sensor, pH, level sensor. So some things are general, some are very specific of the design. For instance, this client requests a biological indicator inside the fermenter and in all the port to facilitate the validation purpose. So in this case also, there is also in all the lines biological inline indicator. So they can do also the validation very good because they will see all the sterilization problem even from this stage. So here one mammalian cell sample. So here, you know, very important the perfusion system. So here also we developed different sample actually for ATF bioreactor when you can control glucose, glutamate, lactose, etc. in the lactate in the process, and to do it in a continuum mode. So see, you can see some sample here. Very complex bioreactor, a lot of sensor, CO2, glutamine, glutamate, lactose, lactate, glucose, etc. Different level of aeration, microsphere, macrosphere. So very complex bioreactor. Here, pH control with the CO2. Here, DO cascade control. When you can control, as explained, with microsphere, or you want to put the air or oxygen into the surface. So all these have a very challenge in the term of automation that also we can follow. So you can see the P&D, what a big complex look. So also we are involved in conjugation vaccines. That is critical for a lot of applications. You have pneumo, for instance, hip, meningitis, ACWY, so X now also. So we also are developing a skip for do polysaccharide fragmentation, polysaccharide modification, and protein polysaccharide conjugation. Also based on the quality attribute of the client. So here you have one, in the previous you have one for fragmentation, and this is for conjugation. So everything integrating with the chemical reaction plus cross-flow filtration. So you can see all the integrated. This is in particular for hip production. So you can see all the integration. Here you can see from the top how everything is integrated. Here they request for the conjugation half glass, half stainless steel because they want to see what is happening in the chemical reaction. Also, we are developing in such a way. Here, another example of the cross-flow integrated. This is very new from the 2018, so April, so 1 year from now. Here you can see another example when you make one chemical reaction in the reactor. After the chemical reaction, you made the diafiltration process in the same reactor. And this is a sample of the integration with the hybrid cyst because they have the addition of the buffer with back. So also we are involved in the retrofitting. So, you know, several companies don't have enough budget to buy new equipment and need to revamping the existing equipment. So also we are involved in take this old equipment that sometimes the vessel, for instance, is stainless steel are good, and we change all the automation and revamp completely the equipment. So at the end you will have almost an old equipment with new brain, but new brain, new sensor, etc. It's a very difficult work, but we are having a lot of experience in retrofitting. Not everybody like to do this kind of job. So here you can see one example, old panel, when they use the actuator with the hand. So this is nobody use it today. So, and you have a SCADA panel, so a new completely panel example. This one we did also the last year. So also, as explained at the beginning, we do what involved in cleaning in place station. Different model. So if you need mobile CIP for clean a particular vessel, or you want a central CIP or SIP, or you have only the pump to do the cleaning. So also this we can do. Or you want to clean the completely facility. You want to manage central CIP station. Also several equipment at the same time also. So you can see some picture here with the single vessel, two vessel, one for reaction preparation, reagents preparation, the other for water for injection, etc. And also, as explained at the beginning, we have also kill vessel. That is also mandatory when you are working with the BL2 or BL3 biosafety facility. So you need to kill the waste. And so we have several examples here. This is an in-batch system. You can see 2,300 liters for vessel. Also for CIP, this is one of the last we made for the recombinant facility. So one continuous also. We have the version of continuous that saves a lot of energy. So in this, you will continuously heating. And at the end, we have the process integration. So you have a facility, you want to integrate the process. This is, for example, one example for meningitis, pneumo, and typhoid vaccine, polysaccharide. So one customer wants to have per campaign production these three antigens, and we develop the completely process flow and integrate everything in one SCADA. So you can see some integration example here from top. And this will be a lot of benefit. So you will have a common process flow diagram to ease the campaign production chain. You will have everything interconnected. You can integrate the process analytical technology as a part of your process because new sensors that was not developed in the past now are available. You can do this as a high-scale production, depending on your possibility. If you have a distributed control system of the PCS7 to monitoring also, we development the SCADA and the automation. We can customize the recipe. So I want to do this, or I don't want to do this also we can integrate. We also perform the continuous automated process if you want. And all the safety concerns, as I explained, also is integrated in the approach, along with the CIP and SIP process. So, like a summary, we can be involved in process technology. We can be in process technology when we can give you a skip model system. So we will develop everything in a skip mode. So you have a skip for fermenter or for cross flow or for media, etc. This will be enable you to do a continuous process. We can do, is everything going good, fast delivery time. In the high scale, as explained you, we cover till 15, 1.5 cubic meter, fully customized with the pre-designed sequence basis of your application. And also if you need equipment retrofitting along with this, also we can do it. In the process control perspectives, okay, we can upgrade your equipment or put new equipment with the new sensor available, like glucose or lactate or glutamate or any new of the sensors that are development. And also we can do a fully automatic facility when you can only touch a button and automate all the operation. So this is my presentation. As explained, I have a final video just to show you most of the things I tell here, but in the video you will see even, it's only 5 minutes. So can you put the video? Thank you. If you have any question, me also and Benki also can reply any question.
Thank you, Daniel, for your presentation. We have a concern as a company, as a client, because normally the headquarters, technical assistance, engineering are not local, you know. As we see, you are from India, you are starting a new business plan here in America, but we are worried about how you will deal with technical assistance and supplying material for equipment, and well, the strategy that you will follow to satisfy that need for us. Thank you.
Okay, in this case, for Brazil market, we are having a deal with the MSM technology. So Mario is here. And the idea is to train the engineering of MSM technology in India. And they have been here based on Brazil, and they can be involved in of the after sale process. So if you have any problem, you will have training people here. Also, Mario is the idea to have some stop of the main spare part regarding with the process we can be sell, and you will have an immediately response because he is in Campinas, so very near. And if there is something complex that they can, very complex problem that they cannot solve, we will send someone from India. This is the idea. Mario even can explain better his idea. And the idea is continue training of the Brazilian engineering he has here in Bangalore, in India. And also, along with the training, the people will receive in the different company. So if we have a business with Biomanguinhos, we will train also the people from engineering and process from Biomanguinhos to put in an example. So this is an idea we are starting now with the first project, as you know. And this is our scope till the moment. Mario also can speak, even can speak in Portuguese.
Yes. Ok. Em complemento ao que o
Daniel mentioned, MS technology is in Brazil for more than 20 years, and our expertise is in after-sales support. We have a contract with big companies always focusing on after sales, because what we understand in Brazil is that high technology companies come here, provide equipment, and then there is a lack of support after sales. So whenever there's a problem, may it be with a valve or a probe or a sensor, there is the need of receiving a technician or an engineer from Europe or the United States or India to solve simple problems. Our idea is the opposite. Our project is there for 20 years, and we have systematic trainings of our engineers on the headquarters, and not only components but also automation, so that whenever there's a problem, it is solved here in Brazil as soon as possible. So this is our proposal, this is our goal, and we've been quite successful with the companies that we work in Brazil. It's just a few. We work with only 3 companies in biopharmaceuticals, and the idea is work with few companies so that we have high efficient services, okay? That's it.
Okay. Any other question?
Mais alguma pergunta?
Okay. Obrigado.
Pessoal, agora teremos intervalo e
Break, so at 10:40 we'll start with the session of Biomanguinhos assets and then the pit session. Thank you for your presence. We'll see you soon.
Bom dia a todos. É um prazer receber.
Good morning, everyone. It's a pleasure to receive you here for our session about the assets of Biomanguinhos within the Innovation Hub panel. I'd like to present Gisele Albuquerque Chades for you. She is the manager of the pilot plant project at Biomanguinhos. Good morning. Thank you. I feel at home here. I'd like to thank all of you for being here. I see lots of friendly faces here. And I want to speak about the importance of you being here because for the first time we are presenting our pilot plant in a professional way. We are starting this out. We will present some advances that we had during these times, and I know we are all very proud of this, especially because this comes from public resources. It is a public initiative, and I'd like to thank the organization of this parallel event, especially Gisele Correio, Vivian Ribeiro, and André, because they really made a big effort. And we made a joke, we said this is like Rock in Rio because we have the world stage and the sunset stage. This is the sunset stage and there's a lot of people here. We weren't expecting such a big audience and we didn't expect so many people. So next year we will rethink the event because this dynamic has been very interesting, this one-on-one dynamic. We've been able to have many conversations, so congratulations to Gisele, Vivian, and André who organized this event. And as I said, it's great to be here. I've been at Fiocruz for 26 years, and I'm going to present this initiative and this asset that is so important for us. We're very proud of it. This is the pilot plant. It's at the Henrique Pena Center at the Fiocruz campus. It's a new pavilion that's just been open, and it's been conceived with the idea of vertical production plants. So for those of you who are taking photos, I can make the presentation available at the end. There's no problem. It will be available for the organizers, Vivian and André. So we really want to exchange this information, so feel free to continue this conversation. So these production plants have this vertical model where on the first floor we have the production of biopharmaceuticals. The fifth floor we have the production of reactives for diagnosis. The third floor, the pilot plant of Biomanguinhos. And the second and fourth floor, the technical floors. In the fourth floor, we have the production of culture medias and solutions that supply our production of the first and third floors. And the transference of these means is made through pipelines through gravity. So in the third floor, we have the production of the pilot plant, and we have about 2,700 square meters of controlled areas. And about 80 to 85% of this area, these are classified areas. So it's a big challenge to maintain all of this space. I see Marcelo from Anvisa here. I say we're going to have a big conversation about this. Our quality director is speaking to Anvisa about defining our regulatory strategy. These are controlled and classified areas, and they are divided into 3 production areas, let's say. This platform of primary production and prokaryotics, so active pharmaceutical ingredients from the cell until the active concentrate, and the primary production in eukaryotic cells, and a whole area of final processing, secondary activities. So it's the whole line of production, the beginning until the packaging itself. These are all specific plants with an air conditioning system, circulation system. There are like 3 production plants in these floors. So we bring this concept from the beginning of multi-purpose plants conceived according to and following the good practices for manufacturing, and it follows all of the requirements for good lab practices and biosafety, working with segregated plants, as I've showed, with the concept of transfer of culture media by pipeline. And these pipelines work with this cleaning, engineering, and sterilization in the location itself where the transferring happens. We have closed systems, so discardable systems, single-use systems. We have the purified water production system and WFI in the plants. So the building supplies in an individualized way the first floor, the third floor, the fifth floor, as I will show in this sequence. And the air conditioning system was designed according to the requirements of the World Health Organization. I'm old now, but when the plant was being built and conceived, we hired a consultant, a specialist from the WHO. She analyzed the whole air conditioning system when we were just starting the design to follow all of these concepts and requirements.
Sim, eu vou demonstrar pouquinho à frente.
I have a sequence of slides about the plants. I hope this doesn't get boring, but Eduardo yesterday from Biominas said that sometimes we get too much into this technical part and it can get a little bit boring, but I need to get into this a little bit, and after this we can schedule meetings and get into the details. So the plants are segregated. We have this B face, 3,200 square meters of controlled areas. Most of them are classified, and they work in the prokaryote platform, and then the A side of the building that divides the production areas, and the eukaryote platform, and the final processing area. So looking at the eukaryotic area, we are going to offer services internally for the final processing platforms and eukaryotics. So the production here of IFA and eukaryotics, it has a separate access that goes through a access hallway, and then you have the support area and the reaction area, or the purification area that are independent. You have this medium central hallway where you have access to the segregated plants, then the operator of this support area, purification area. They are all in independent areas. They conduct their operations in the rooms and exit through this external hallway. So we respect this flow. The access is independent for these areas, for all areas, whether it is support, bioreaction, purification. They have independent exits to the exit hallway. In the same way, the plant for final processing has the independent access through a central hallway that gives access to these areas: the support area, the formulation area, or the area of cleaning of recipients for the primary packaging area, or this final packaging area. These are independent operators. These are segregated areas, both physically. And so for the end of the process of formulation or packaging, they exit through this exit hallway in order to enter the final processing area. We have the access for the secondary packaging area. We have this control and access area and the machinery area. and the secondary packaging area that has independent access through the hall of access. This is a different idea. It's totally segregated of the rest of the primary packaging area.
So you could have the overview, but we have all the details of the flows and processes being described in memos and also flow plans. Here are some brief images. The system to produce water in the building, working to all plants independently. The conception is of overcapacity. We have a production in strong volumes. We produce per hour 42 cubic meters of purified water, 18 cubic meters of WFI per hour, and we also have the steam generator that in routine with the productions under operation, we estimate that the use of this water system would be about 30% of the capacity of production. We work with the safety limits regarding that strongly in all the conception in the pilot plant, and especially in the first floor, so we know that in case of undersupply of water or any problems in supply to the building, we have the safety to ensure the processes, because many of them we can work in batch process or feeding processes or continuous process. Some are continuous and you cannot interrupt any time. We have process of 110, 130 days of production, so we cannot lack water. That's why we have this overcapacity of production and we open with the safety margin. Here are some images. We are in a conditioning phase. Some images just for you to see. We have here the machine and passive reds and the lyophilizer with automated loading and the lyophilizer in the machine area. And this is a room for bioreaction. We have single-use reactors. We work with bags in the end of the process. We know that due to regulatory problems, we see that it's very well used. So we invested in this strategy in the pilot plan. We have also systems for tangential filtering. We have some partners here in the room. We have chromatography systems and all the primary operations for the production in eukaryotes. In Biomanguinhos, we have the conception of matrix work, so the idea and the guidance of our directors is that we have in the pilot plan our experts by platforms, so they will be the focus points of the platforms of bioreaction, lyophilization, secondary operation. So we still count on all the Biomanguinhos experts in innovation and production to give support to the experts in terms of knowledge to any kind of service that comes to the pilot plant. So, for example, we count on 110 doctors, 230 masters, and almost 1,700 workers which could give help and identify and study and research any kind of service that is provided in the pilot plant. And everything was built with an important partnership. Yesterday we had here the managers of FINEP and BNDES, and we thank them a lot for the partnership. Sebrae is a great partner working in the management front of the project directly with Vivian and all of us. We had also funding for the regulation of the pilot plant and risk assessment for the final processing. Everything is built in partnership. So today I am here representing a big team that is working from the matrix to metrology, validation, management, quality. So nothing was built by chance. And we believe till the end of the year we start with the service itself in terms of final processing. We'll be preparing 3 consistency batches, so the projects of transfer of technology in Biomanguinhos, at the end of the year we'll have a team working on site and in the line of final processing, so that in the first semester next year we focus on bioreaction in eukaryotes and also purification, and we follow the work little by little, and all the team will be there, gathered and working for that. Once again, I thank you all for your presence. That's what I had to show.
Obrigada, Gisele, por suas
Thank you,
palavras.
Gisele, for your speech.
Vou abrir no final para as perguntas, ok?
I'll open for questions in the end, okay?
Agora eu gostaria de chamar a doutora Maria de Lourdes Souza Maia, gerente da
Now I would
assessoria
like
de
to invite Maria de
assessoria
Lourdes Souza
clínica de
Maia, who is the
Biomanguinhos.
manager
Seja
of
bem-vinda.
clinical assessorship for
Olha meu
Biomanguinhos.
olhar,
Welcome.
gente. Eu sou
Look at
Lurdinha.
me, girls.
Ali está a Letícia
I am
Kegler, que ainda agora eu falei com
Lurdinha,
ela,
there
olha,
is
tu vai
Letícia
também me
Kegler,
apresentar
and I
umas
said,
duas. Ela,
well,
pelo amor de
two
Deus,
of them
você
will present
tá
now. And
dizendo
she said,
isso
oh
aqui,
my God, you're
mas
saying
a
that here,
gente
but
tem na
we
vida,
have
a
in
gente
our
tem
lives
que saber
the
suportar
goal
as
of
pressões e
supporting
construir
pressure
andando, né?
and
Você troca as rodas
work
no
and
carro com
walk
ele
all at the
andando
same
também.
time.
E
We
é assim
change
que
the
a
tires
gente
when the car
construiu
is
a assessoria clínica. Começou
running.
com o Dr.
So
Reinaldo
that's how we
Menezes, a quem
started
a
our
gente
assessorship. We
homenageia
started with
neste
Reinaldo
momento,
Menezes,
e depois
and
eram
our
5
homage to
pessoas.
him
Eu não
now,
digo
and
quantos
later
somos
on
hoje,
5
porque tem
people.
pessoas
I
da
won't
diretoria
say
aqui
the
que vai dizer,
number
"Ah,
today
vocês
because there
cresceram
will be
muito,
people here saying,
etc.
no, that's
e tal,
more,
né?
and so
Mas
on and on. And
todo esse trabalho,
all this
ele
work
é feito
is
com a participação,
carried out
estou certa,
with
eu não sei passar.
the participation.
É atrás? Não, eu estou usando até a certinha. Travou. Tá. Tá travado. Então é um trabalho feito por
So
uma
this
equipe,
is a study, a work carried out by a multidisciplinary team.
Eu apertei no meio e aí travou. Não, é porque eu achei que tinha laser. Aí eu
I'm
fui
sorry, I
procurar o
thought there was
com-
laser here, so I was looking for the laser pointer.
Ah, tá bom. Eu tenho 15 minutos, né?
I have
Você,
15
então,
minutes,
toque quando
right? So
quiser
you
10.
tell me when I have
Ok.
spent 10 minutes.
Pode ser? Pronto. Então, essa assessoria clínica,
So this
desde
clinical
2005, ela tem
advisory since
como
2005
objetivo de
aims
estar fazendo
at
planejamento
planning
de
and
diferentes estudos clínicos,
creating
né? Pode
different
ser de
clinical trials,
vacinas,
may it be for
biofármacos,
vaccines,
kits
biopharmaceuticals,
diagnósticos, a gente
diagnostic
prepara
kits.
aí.
We
E
prepare
principalmente,
everything.
se
and
pode
mainly,
ser transferência de tecnologia,
If we are talking
pode ser
about technology
desenvolvimento
transfer,
conjunto, a gente
joint
tá sempre aí
development,
trabalhando.
we are always there
E
working together.
monitorando também
And also
ao longo
monitoring
de farmacovigilância, esses
pharmacosurveillance
estudos ou casos
with
que
all the
estejam ocorrendo também.
studies or
Porque o que a
cases
gente quer é a
happening, because
segurança dos
what we
produtos.
want is
E a
the
gente
safety
faz,
of the
portanto,
product, and
estudos
then
pós-marketing
we
também.
study, we
E
have
tudo isso
aftermarket
seguindo
studying as well.
o
And
regulatório da
always
nossa querida e
following
amada
the
Anvisa,
regulation
dos
of our
comitês de
surveillance
ética que estão
agents
aí
and
para
the
que a
ethics
gente garanta a
committee,
segurança
so
dos-
that we
do
ensure
nosso
safety
pessoal, né, dos
to
nossos
our
participantes de
personnel
pesquisa.
and all the
Tem uma coisa
participants.
que a gente
There is one thing
coloca, e eu acho que é um dos pontos
that
que
we
deu também
pose,
o
and
êxito.
this is
É
one of
o
our successful
relacionamento
points, that
com
is
as
relations
associações
with
médicas,
medical
né? A gente
associations.
não faz nenhum estudo
We
clínico
have no
sem
clinical
chamar. Se vai fazer
trials
algum que
without
seja
calling everybody. I
envolvendo
mean, if you have
nefrologia,
something involving
Sociedade
nephrology,
Brasileira de
we talk to the
Nefrologia.
Brazilian
E
Society of
diabetes,
Nephrology,
Sociedade
diabetes,
Brasileira
the
de
Brazilian
Diabetes.
Society
E
of
assim a gente
Diabetes.
constrói
And
junto
therefore,
um
we
protocolo,
build together the
discute com
protocol,
eles
we
e tem a
discuss with
participação
them,
deles.
and
Isso
we have their
garante também pra
participation.
gente
It also ensures tranquility in our work. We also have the support to any other epidemiological studies or pharmaceutical states. Everything is our job to do, and also in collaboration in the organization of workshops and also with the prospection for new studies. That was not the slide to be presented. I have to tell you, due to our vice president here, this is the proposal to have the new structure. Then they said, but Lourdinha, it's not like this. And I said, well, I can't do with this. We are just having a coordination of clinical assessorship. That's how we work, so I will have it the way we work today. How do we work today? We have the administrative support, and we have the areas of clinical operation and pharmacosurveillance and access to health, where we have the associations of patients and medical associations. We have the medical scientific, and we have the banks and biobanks. And then we have the project leaders, monitors, quality management. I tell you, I miss my PowerPoint, but we also have the investigational product. When you said that we will have the 3 batches, I asked there, so my product, my investigational product may be produced there soon, right? I don't have to fight with the production, to stop the production, to prepare the batches, the investigational batches. That this is the greatest challenge we face today. We make produce to sell or produce for clinical trials. Georgia knows that's the reality. Wonderful, but here, well, it's not mine, it's of our every research, because we are here to say what are the capacities of Biomanguinhos to be offered to anyone who wants to use it. So this is what I have just talked to you, that as clean, since the scientific, medical scientific and clinical operations and planning, monitoring, administration, and even pharmacosurveillance. But how can we have clinical research in the unified health system? This is the big point, and this was the greatest challenge. I told you that we tried to find the medical associations, but the medical associations just not that we have any noise in the communication, so that we have influencers with us and so on and on. But what about execution? How do we do that? So we search for partnership with universities. Probably someone here doesn't like it, but today this is the management of university hospitals. So we searched for universities and the municipal secretaries and many others that we could have, because this is where we'll find our target. If I have a clinical trial with the vaccines, where can I find my participants? I need to have 5,000, 10,000 participants. Where do I find them? I find them in this health unit. And how do I go there? We need to prepare the units to be part of the treatment. We need to give qualification. If you have a doctor that is assistentialist, but his mind to be changed into a researcher is difficult, because he believes it's not his job. He doesn't have to do that. And then we have all the qualification and training for those how can we deal with the community? Because they will say that they are being treated like guinea pigs, so we need to work with the boards of health, like the municipal board of health, the district board of health, neighbor associations. And then we can have this situation. This is what we could do in the country so far. Everything in red is states in Brazil where we could have clinical trials using this base that I presented to you of the public services, the health units, going there one by one, talking to the municipal secretaries, talking to the ethics committees, and everything according to the standards of clinical trials. And here are the current ones where we are today. We are now developing in these states clinical trials, clinical research, and now we'll go to Mato Grosso do Sul. Pará will have another research because we already have one, and now we are going to the metropolitan area of Rio de Janeiro. Yesterday we went to Duque de Caxias, São João de Meriti, and tomorrow we'll deal with the São Gonçalo. This is such a rich process of talking to the Secretary of Health, and I can give an example. On the 3rd, we were here on a meeting discussing the diabetic patient, but what does it have to do with that? It is that we have a biopharmaceutical study for diabetic ulcer with a product that is technology transfer, that is promising and will avoid amputations. So we cannot include participants because when we get to the research site, the patient is already there for amputation. Brazil is only cutting, and I see that there is a problem between the vascular surgeon and the endocrinologist, because when the surgeon receives the patient, he has to do the surgery because there's no other way. But the endocrinologist says we need to take care of diabetes. So we gathered everybody in the same room with the National Board of Health and the Secretary of Science and Technology and the National Association of Diabetes. Everybody came to the meeting and we started discussing diabetes in Brazil. And we ended up with all the municipal secretaries going step by step in the training of people. So when we have research within the unified health system and public sector, we need to spread our point of view. We have to know that at that moment, as a person, a citizen, what can we do to contribute? We may have scientific knowledge and bringing self-esteem and looking at how the system is at the moment, and this is what we are doing. So these are the permanent centers we have here in Rio. These ones are ready for the research. Just go there and start working. We have one in Tijuca, another one in Fiocruz, and another one in Santa Cruz. But in Santa Cruz and in Manguinhos, we already have a center for sample processing because 2 hours to transport the sample is too much. If we get a traffic jam, it's a problem. So we need to be there working. So these are all the sites that we have, even the international ones. Here I'm showing some of our launches. The first one was that one, first, second, and third of our new units. And in 2006, from 2006 to now, we finished 39 clinical trials in phases 1, 2, 3, and 4 with vaccines, biopharmaceutical, diagnostic kits, and these are under development. So I believe now we have 7. These are the ones we are developing.
Aqui é porque a gente tem, além do biorepositório, foi o primeiro
Aside
biobanco,
from the biorepository, we were the first biobank accredited by CONEP, in Fiocruz, that is accredited now. It is accredited, right? That's how they say it.
Aqui o biobanco.
So here's the
Dois
biobank,
sistemas que
and
nós
we have
criamos.
two systems created.
G-Clean,
One
que é o sistema de
is
gerenciamento,
G-Clean,
ele é
that is the
maravilhoso.
management system.
Você
It's
tá com
wonderful.
insônia,
If you
no
can't
computador,
sleep,
vai
you turn
lá
on your
e
computer,
olha
go
lá
there,
Paraíba,
and look how it
qual
is
era a
in
meta que eles
Paraíba
tinham
and what was the
por
goal
dia,
they
por
had
semana, por
by
mês,
day,
e
by
fez por
week,
mês.
by month, if
Aí
they
vai no
did their
WhatsApp
work
e
or
ela
not.
sabe,
And then
ó,
you go there
vocês estão
to
com atraso
WhatsApp
aí, o
and
que
say,
é que tá
hey, you are
Mas por que
delayed,
isso
what's
o gestor
happening?
tem que fazer?
Because this is
Pra
something the manager
identificar a
has to
causa
do to
raiz,
identify the
gente.
root
Se a gente
cause.
quiser caminhar,
If
a
we
gente
want
tem
to
que saber
follow the
responder o porquê
pathway, we
da
need
causa
to know
raiz.
how to
E aí a
answer
gente
why.
consegue
This is the root of the problem, and then we can overcome problems day by day. And then we have Bioform, that was Sepro who did
que
it
é
to
pras
us
fichas clínicas,
for
todo
the
em português.
clinical
Todo em
forms,
português.
and everything is in
trabalhar no setor
Portuguese.
público,
If
dá
I
pra
have to work in the public sector,
sistema em
I,
inglês?
can I have a system that is in English?
Tem que ser em
No, it's
português
impossible.
mesmo,
It has
e
to
pode
be in
ser
Portuguese.
customizado.
And it can be
Você faz também.
customized. You
Então,
can
a
also
gente
do
tem
that.
esse. E a
We have this one and
companheira,
surveillance,
rápido,
pharmacovigilance.
levanta,
Come on,
fala,
come
ela
here
é
quickly.
Eu era
Because I worked there. Now I was caught by surprise here.
É uma atribuição da
Because
também.
this is also an
E
attribution
funciona
of
numa lógica
ASCLEAN,
diferente
and it
das
works in a
clínicas.
different
ASCLEAN
logic
funciona,
from ASCLEAN,
é,
because
planejando ensaio
ASCLEAN
clínico, executando
plans
ensaio
clinical
clínico, farmacovigilância
trials, and
rotina,
pharmacovigilance is
atribuição regulatória,
routine
né,
and
parte
regulation.
E a atribuição principal da farmacovigilância
And the
é segurança do
main
paciente.
attribution of
o que que a gente
pharmacovigilance
faz,
is
ou
the
fazia,
safety of
né,
the
que agora eu não
patient.
faço
So
mais.
what we
A
did
gente monitora eventos
was that
diversos.
we
A
monitor
entrada dos
adverse
biofármacos, ela muda
events,
pouco essa
and the
lógica, porque a gente
biopharmaceuticals
passa
change this
a ter
logic a little bit
contato
because we
com o
start
paciente
having
por
some
conta de
contact with the
programa de
patients
suporte,
due
que
to
antes
some
a gente
support
recebia
programs,
mais
because before
via
we
sistema de
received
atendimento ao
more
cliente.
things by
Então, é
the
volume
service of
maior de eventos que
patient
a gente
care, so we
avalia,
have more
mas a
events
intenção
now to
principal é
assess,
avaliar
but the main
continuamente
goal is to have
o
continuous
perfil de risco e
assessment of
segurança
the
do
risk and
produto.
safety profile of the product.
A seguinte é minha. Eu não sou médica, eu sou
Well, I'm
farmacêutica,
not a
mas
doctor,
a
I'm a
gente tem dentro das
pharmaceutical
clínicas hoje 7
pharmacist.
médicos, a gente tem
So
uma
we
área
have
médico-científica,
7
tem dois
doctors, we
médicos que
have a
são
medical
da
scientific
farmacovigilância
area, we have
também.
2 doctors from
E
pharmacovigilance,
a área
and
médico-científica
the
dá suporte
medical scientific
ao
area
desenvolvimento de
supports the
protocolo,
development
avaliação
of
de novas
protocols and
a gente
evaluation of new
representa as
technologies.
clínicas,
We
relatório da
represent
Anvisa,
ASCLEAN
a
and
gente
the
dá
national
suporte ao VDTEC na
surveillance,
avaliação de
and
novos
we
produtos,
support in the evaluation of new
faz
products.
monitoria
We
médica
monitor
nos estudos clínicos.
doctors in clinical trials.
Acho que eu consegui cobrir tudo.
I think I
Protocolo, dá
mentioned
suporte
everything.
em
We
estatística
create
no
protocols
cálculo
and
de
support
tamanho
statistics
amostral,
in
porque, né,
the
tem variáveis
calculation of
clínicas
the
que entram no
samples because
cálculo.
we have clinical variables in the calculation, and that's it.
Obrigada. E esse aqui, gente, é
So
pouco das
here is a
parcerias
little
que a
bit
gente
of the
tem, né? Isso
partnerships
não se
that we
constrói de um
have.
dia
We
pro
don't
outro.
have this
Amanhã
from
eu
night
tenho
to
uma
day.
reunião inteira com a
Tomorrow I
Sociedade
have
Brasileira
a
de
big
Reumatologia,
meeting with the
por
Brazilian
conta de
Society of
produtos,
Rheumatology
mas
due to
agora
products, but
são 3 em 1. Tem
we
com a associação de
have
pacientes
also a meeting with the
reumatológicos.
Association
E tem uma
of
coisa,
Rheumatologic
Marcelo, que você vai
Patients,
gostar.
and Marcelo,
Eu
there's
vi
something you will
a
enjoy.
Sobral,
I
Flávia
saw
Sobral,
Flavia
2 anos
Sobral
atrás
2
falando
years
que
ago
a FDA
talking
estava
about
botando
the FDA, and
pra
they were
discutir
putting
pouco de
patients to
protocolo.
discuss
Nós já estamos
protocols.
fazendo
We are
isso,
already
tá?
doing that.
Amanhã a gente vai ter
Tomorrow
pacientes
we will have
discutindo
patients
protocolo, e
discussing
o de Hansen,
protocols,
a gente já tem o
and
Mohan também
in the
trabalhando
Hansen
com a
we'll
gente.
have Mohan
Quem
working with
faz
us.
aqui, eu dou conselho.
I'll give you an
Eles
advice to
têm uma
you.
visão
Have that,
que a
because
gente
they
não
have
tem.
a view
E
that
isso
we
ajuda
don't,
muito,
and it
muito
helps
mesmo,
a
tá?
lot,
Então,
really
esse
a
daí,
lot.
a publicações que a
And
gente tem que fazer.
The publications that we have to make, the last one that I'm very proud of was by Dr. Reinaldo, and we were able to do this. It was 8 years working on this vaccine, and now it's been 10 years. And for 60 or 70, we had been collecting the blood, and now we can give the answer about whether the fractioned vaccination works for 10 years or not. And this is our team. So that's why I said I wasn't going to speak so much. This is a permanent team, and they are all managers. You can't just pat people on the back. You need to build things together, show the paths that can be followed. Yesterday I turned 69 years old, so I'm thinking about retiring, and we need to prepare the team so that everything can continue. This is only possible thanks to this team. If you're making an ugly face, you won't be with us tomorrow, but you can see our team here. Thank you so much. Yes, please, if you want to ask any questions. Does anyone have questions? We only have 5 minutes, but we have the microphones here. Just raise your hand. I'm not going to make a question, it's just a comment. Lourdinha, I just missed in your presentation to speak about the partnership with the surveillance agency. You spoke of the Ministry of Health, but not the surveillance agency, so I was a little bit jealous, you know? I cannot translate if they are not speaking on the microphone. Gisele, I enjoyed your presentation. I hope your project is very successful. I really trust all of the Biomanguinhos projects. It's important for us to have this practice of installing the products to avoid the competitors. Rosane asked me a question before we got here about this pilot plant and the CMPF. It's very important for all of the phases of manufacturing medication, but not for clinical trials. So I would recommend this certification, even though it's not necessary for manufacturing medication. I do recommend you receive a visit from the surveillance agency here in Rio or in the country, because if you're going to present your work, your product that was produced in that plant to the surveillance agencies, they will not question your product. They will not ask, oh, this was produced in a space that did not have this specific, that was not accredited properly. So if you are starting now and you seek the support of the agency that regulates this, it's good to seek out these partnerships, work with the surveillance agency. I know you didn't present so many plants. You should present all of the details involved in this in some meetings, even before you call the surveillance agency over to inspect the physical space as a whole. I did have some doubts about some of the text you had there, like when you speak of it being dedicated to a HVC and physically too, in the representation you put there, I couldn't really see it very well because you use the same colors. So in an area that is dedicated to something, can it have the air supply or the air exchange with a different area that theoretically is segregated? So it's good to question and to check all of these numbers and issues, because it wasn't so clear for me to understand this in your presentation. But I did enjoy it. I think you're following the right path. But now, because you're just getting started, try to really look at all the details in the best way possible. Be perfectionists about it. We're here to help you regarding the certifications and to support this project because I know it's going to be very good for Brazil's future. Congratulations, and I wish you success. This was very good advice. The surveillance agency was always and will always be a strong partner. As you said, we're always together in this and we have this public commitment. The colors in the presentation are just for representation purposes. They don't represent our engineering plants or represent the flows of the process or anything like that. They were not organized for this. These are not technical diagrams in the slides, okay? The more technical plans and diagrams are being prepared so we can present this to the surveillance agency and be discussing this actively and strategically so that we can work with the technicians of the surveillance agency. I'm not a specialist in this. We're making this presentation for Alessandro and the engineering team that works closely with us, and we will speak about this in great detail. I'm sure we will build this with the surveillance agency. It's the first pilot plant in Brazil, and I know you will help us in this construction process because it's new for everyone, the legislation. We're looking at what already exists abroad, which is a bit different from what exists here, but we're studying this so we can work together and build this regulational issue and construction. And thank you for your advice. We're very committed as Brazilians and as public employees to build this well, and thank you for your advice. Any comments? Thank you. Thank you, everyone. We will have a 5-minute break until our pitching session. Thank you and see you soon. board members once again. I'd just like to say we had to make a replacement because Eduardo from Biominas could not be here because he had something urgent today, a health issue in his family, so he had to go back to Belo Horizonte. And so to replace him, we have found Zenia Laura. She is a representative of the Ministry of Health, of the biotechnological area of SICTI, which is the Secretariat of our Health Ministry. It's a pleasure to have you here with us. We have João Miguel as well, who is an advisor in the tech and innovation area in Biocruz, and our vice directory in technological innovation could also not be here because of issues with their flight. So Marceli could not be here. Thank you for your presence, and thanks for all of you for being here. And let's start these presentations. Starting this order, we have Wesley's presentation about RNA loading in lysosomal diseases. So you have 5 minutes to make your presentation, and I will make a signal when you have to finish. Have a good presentation. Thank you. First of all, I'd like to thank the opportunity to present our pitching idea, to pitch our idea. My work is based on nanotechnology therapies related to genetic material. And our project is using nanoparticles, liposomal nanoparticles associated to genetic material for other forms of therapy. So I'm going to present the model. It's a model for the Gaucher disease. It's a model that has an interesting use because it has some technological advantages. I don't know if this is working.
Só um destaque para os apresentadores.
Please don't press the middle buttons because you can interfere with the communication. It's just front and back. But the laser pointer doesn't work because the screen is too light. See, you can see it here, but not on the screen. So let's start over. Well, I'm going to present this idea that we've been thinking about it for a while now. Gaucher disease is a recessive genetic disease, so for you to manifest this, you have to have two deficient alleli of a specific deficiency that works inside a physiological system of the lysosome. So any person who has a normal allele of the disease, if this gets in touch with some substrate that has lipids that compose this substrate, you can reutilize the parts of this substrate for cellular purposes. If you have two deficient alleles of this disease, they cannot be degraded. These lipids start accumulating in the cells that should be degrading these substrates. The main cell responsible here are monocytes, and they are usually the first cells to clean detritus from our body. If you have macrophage, you might have a problem related here. If you have the Gaucher disease, you might have issues with your red blood cells because of this immune issue. And in the most serious forms, your nervous system will be affected, and this causes mortality. So how is the treatment conducted today? Usually you will have a recombinant protein. This enzyme that the person cannot produce in an efficient way is produced in the lab, and you conjugate in this enzyme groups of sugars to try to force the macrophage to swallow this enzyme, and this enzyme eventually escapes from this phagocytosis process and can arrive at the lysosome. The issue with this technique is that it is very expensive. You can have to make the protein, place the sugar, and then wait for the physiology of the macrophage to phagocyte. So Biomanguinhos itself produces this recombinant enzyme. Our idea is not to replace the enzyme. We want to work with the RNA here. Why RNA and not DNA? Because any genetic therapy has many issues and side effects when it is applied. If you're going to use the DNA, it could integrate into the cell and become a tumor. So if we use RNA, it's temporary. So returning the RNA here, these macrophages can phagocyte the RNA, translate this and create the protein inside themselves, and taking them to the lysosome. So these proteins can have different destination, but our idea is to use the RNA. The issue is that it's very unstable. So that's where we get into the nanoparticles. Protected inside the nanoparticles, the RNA is stable in the blood flow, and you can apply it. And it has high rotation in the blood through this kind of formulation. This is the formulation that I created in the last years of my doctorate degree. And this is not such a new idea. The industries in the US and Europe have been working with this for some time. Moderna has this same system, but it uses the modified RNAs, and it has its own particles. But of course the idea is the same, protecting the RNA, integrating it to the cell, and the cell producing the intracellular, if it's a membrane protein or if it has to be secreted. For you to have an idea, I didn't find the most recent data, but in 2017 Moderna received investment of 6.9 billion. There's a point missing here. And other places are doing the same kind of therapies with the RNA. The problem of using the liposomes is that if I have my particle, it meets the deficiency of its use. If I combine the RNA and lysosome, the macrophage will consume this. For immunological responses that can generate vaccines, or for putting this inside the lysosome so you can have the protective effect against the effects of Gaucher's disease. I think in a general way, all presentations finish with the logo of the company or something like that to show the idea. For me, it's important to bring this idea along. If you have 6.9 billion ideas in one idea, it's because there is a profit in this idea, and nobody's doing this in Brazil. Nobody is investing in this messenger RNA. I know there are some RNA works around here, but not messenger RNA. It's important to remember that these people suffer with these diseases, so we need to bring this forward. Thank you for your presentation, Wesley.
Now the next presenter is Andrea, who will bring the project of iBench. Okay, good morning. Thank you for the opportunity to introduce to you iBench. I am a researcher and one of the founders of iBench. Our goal is to bring solutions to the science world. Me and my partner, Deborah, lived in the academy for many years, and we brought iBench with the idea of bringing our own experience in science and university. So iBench came with the proposal of having this experience that I had in the academy, working many years in the development, developing studies in melanoma. I worked many years with melanoma, and when I went to the United States for my sandwich doctorate, I could see how easy and fast it would be to develop research. When I came back to Brazil, I said, okay, something has to change. Something's wrong. We need to change it. So talking to my colleagues, I understood that everybody faced the same problem as me. So we had problems in research and transducent because we didn't have the reagents to be easy for the development of the research. So the risk of the project was big, so it could even make the project change completely. So iBench tried to involve science and technology so that we could present to the researcher that they could work in science the best way they could. That is the best thing to be done. So our proposal is that iBench is a startup. This is our first product, iBench Market. That is a marketplace to connect science to different suppliers, finding their lab products easily, and the researcher may find easily a product they are searching for on the market for a better price or with a faster freight and with important characteristics for the project to be developed. So we see that finding products is something that doesn't happen only in the university. We have research institutes or hospitals in need of the product to develop their own research. So for you to have an idea today, the public investment in research and development is about 18 billion reais in Brazil. It can be accounted to up to 15 billion years if we take into consideration private investments, and it's important. We need to work for that to be used the best way. So I bring to you our first product of iBench Market, our marketplace that is under operation. We launched it in February 2019, and we can reach now the city of Rio. We'll soon have the national launch. We have our first monetization. Our main source is the split of payment, where we have a small percentage coming to iBench through the product. So the researcher doesn't pay anything. The supplier pays with the contract they have with us. This way, we may think that the marketplaces in Brazil are growing a lot. It's an important tool, a very promising one, and we are thinking that it is a market that we have $300 million in Brazil in terms of lab product purchase, and we want to reach 2% of this market. This is the iBench team, me, Deborah, and Bruna coming from basic research, and then she took her MBA in project management. We have two wonderful guys working with us to change everything into a product. We truly believe we can change science in something very important for scientific development. One more data to you, iBench is being accelerated by a startup program in Brazil. We worked with BNDES, and now among the 1,700 startup companies evaluated, we were in 57th place. So we are very proud of making it happen and being a company that is from scientists to help scientists in Brazil.
Obrigado, André, pela apresentação.
Thank you, Andrea, for your presentation. Just to remind you, comments will be the end of the presentations, okay? Now, next presentation is Karina and Natila. 5 minutes.
Quando é que passa aqui? Então, é só... Quer revezar, só tem um. Sim, claro. Não perto do meio, só pra frente. Só pra frente. Tá, se precisar botar alguma coisa pra trás, mas acho que não é seu caso. Beleza. Tá? Bom dia, pessoal.
Good morning, everybody. I'm Atila, biologist and researcher, and recently I finished my MBA in development of new business. I'm Karina, I'm also a biologist, and I spent the last 10 years in a traditional patent office helping big companies. Today I am a partner of Venturi IP, that is a partner of IPUSH project. Last year, Brazilians were responsible for only 20% of the patent files in Brazil, and it's even more important if in 2017 we had 4 million startups in Brazil. So the reasons to explain this low adherence of Brazilians to the patent system is the cost of processing the patent and the file itself. So you have an initial peak in the graph showing that 25% of everything spent in processing comes in time zero, which are the services of search and the filing of the process, the project. But then we dilute the other 75% over the processing and the end of the term of the patent. So we divide it into 15 to 20 years. Mainly with this initial peak, we can see that in fact the bottleneck in terms of finances to enter the system are the initial steps of filing a patent. In this sense, we created IPUSH. In fact, it will try to decrease the initial bottleneck to enter the system, and it will work as a bridge between research and the IP systems. It will come as a solution for the problem of new ideas entering the system. It will also give access to the processes that are so expensive, the services of search and writing, and without compromising quality, because this stage is so important and essential for the good protection. And how to make it viable? Our business model is established by partnership, and we'll find biotech projects to access the acceptance and select the projects for the program. As selectors, we'll write the patent and give support to the filing process both here and abroad. Thus, we'll offer service in a very good price, at cost price. We won't charge for the lawyers, and we'll only charge the clients with the official prices. So the DNA of IPUSH is philanthropy. We want to push the development of biotechnology and bring IP protection, intellectual protection. Venturini IP are partners in the benefit of the marketing strategy, having a stronger brand and with strict relations with investors and international offices. Our team comprises 6 professionals involved in this project in IPUSH, and it's a multidisciplinary project. We can work with 10 projects yearly. That is the project we can work without any external investments, but with the financing of IPUSH, we could scale the project and work with much more patent files in science. So the main goal here is to present to you an invitation so that inventors and possible investors gather us in the idea and contribute to the development of biotechnology and the development of technologies in Brazil. So welcome you all, and thank you for your attention. Thank you.
Aqui, ó, seu certificado
Obrigada, viu?
Obrigado pela apresentação, pessoal. Obrigado pela apresentação. Agora...
Thank you for your
Próxima
presentation.
apresentação é da
Now,
Maria de
next
Lourdes.
presentation, Maria de Lourdes.
Certinho. Aí é só a visão.
Bom dia a todos,
Good
meu nome é Maria de
morning,
Lourdes
I'm
Magalhães
Maria
e eu
de
vou
Lourdes
apresentar hoje
Magalhães
meu
and I'll
projeto
present my
intitulado
project
Desenvolvimento
called
de novas
Development of
ferramentas
New Molecular
moleculares para
Tools for
detecção e
Antibody
purificação de
Detection and
anticorpos.
Purification.
A importância dos
The
anticorpos
importance
secundários
of
todos nós
secondary
conhecemos.
antibodies,
Eles
we
se ligam aos
know
anticorpos
they
primários
link to
ligados ao
primary
alvo. A
antibodies
principal
and
desvantagem
alpha.
dos
The main
anticorpos secundários
disadvantages
é o seu
is
alto
that
custo
the
produtivo
high
decorrente de
productive
um processo
cost
produtivo
due to the
complexo.
complex production.
Em contrapartida, nós temos
On the
proteínas
other side,
bacterianas
we
como
have
proteína A,
bacterial
G e
proteins
L, que são
like
proteínas
A,
de
G,
custo
and L,
produtivo bem
which
menor,
are
mas que
very
apresentam as suas desvantagens.
cheaper,
Por
but with
exemplo,
the
proteína A não se
disadvantages
liga a
like
alguns
protein
subtipos de
A
IgGs.
not linked to
IgG
some
humana
IgGs
3,
like
esse tipo 3 não é
human
detectado
of
por
subtype
proteína A.
3 that is not detected
Proteína
by
G
protein
tem
B. Protein
espectro
G
de ligação um pouco mais
binds
amplo, mas
a little
apresenta
bit
uma
more,
importante
but there is
desvantagem
an important
que é durante o
disadvantage
processo
that
de
is
purificação de anticorpos
during
utilizando
the
essa proteína,
purification
o processo
process.
de eluição
The
necessita
elution
condições
process needs
ácidas muito
acid
drásticas que muitas vezes
conditions
altera,
that many
desnatura,
times alter or
danifica
denaturate or
a IgG a ser
damage the
purificada.
IgG
O
purified.
mercado de anticorpos
The
está
antibody
em crescente
market
expansão,
is
deve
constantly
atingir 140
growing and
bilhões de
will
dólares
reach
anuais em
$140 billion
2024. E a
in
cada ano, o número de novos
2024.
anticorpos monoclonais
And
entrando no
every
mercado
year, the
é maior.
number of monoclonal
Dessa
antibodies is
forma, a
higher. Thus, the
necessidade de
need
nós obtermos, de nós
of
termos em mãos
obtaining
mais
and taking
ferramentas
into our
moleculares
hands
capazes
more
de
molecular
detectar
tools
todos
to
esses tipos de
detect
IgGs que estão no
any kind of
mercado, não somente
IgGs on the market, and not only detected, but purifying with high efficiency and low cost is quite important. In 2010, a paper was published showing an intracellular protein able to binding IgGs of viral particles and generating the degradation of the virus and bringing intracell immunity, something that wasn't thought before. Our idea was to analyze the protein and take the binding Fc region of the protein to create a chimeric protein that we called Y1902. The molecule is represented here with the linker connected to a streptavidin. And the rigid part, it was patented in Brazil. The international ICT was requested, and the goal of purification and detection of IgG antibodies. The advantages of this protein, first, the production cost that is low, because bacterial production in E. coli, we have a wide range of IgG detection species, even the human of type 3, conjugation to several biotinylated molecules like resins for the production of chromatography solutions for purification, and the main advantage to me so far is the elution process using the chimeric proteins regardless of adding salt. So there is no need of acid conditions during the elution process, thus avoiding the degradation or damaging of IgG to be purified, because the link of IgG is sensible to the presence of salt. When we produce the protein and we test the capacity of detecting IgG, it is capable of detecting in many species, mainly dogs, pigs, humans, and rabbits that is not in the slide. When we compare the capacity of detection of this protein facing monoclonal antibodies, secondary monoclonal antibodies, we see some species better detected by our proteins than secondary commercial antibodies, like human serum. Some other species show a lower binding, but comparable, but we are using one protein to detect all species, and antibodies are species-specific. Who's testing our prototype? Some in vitro diagnostic industries and some labs, institutes, and universities in Brazil. This is our team. The project is developed in partnership between Cinco startups and Santa Catarina and two institutes Senai in Rio to create the chromatography column. And what do we want? We want technical and commercial partnerships to scale the production and distribute the molecule. It's missing one slide here, but our goal is not only to substitute proteins A and G in vitro diagnosis, but also complement their activity in moments of failure of the proteins and also create the chromatography column to purify IgGs to be in the pharmaceutical market. Thank you.
Só para certificado. Aqui o certificado. Obrigado pela apresentação, Maria.
Thank you for your presentation, Maria. Next presentation is by Gabriele. She will speak of biologically derived magnetic nanoparticles.
Tudo certo. Bom dia, meu nome é Gabriela.
Good morning, my name is Gabriele. I am a postdoc. I work with the Labomax group. Our nanoparticles are formed by magnetotactic bacteria. These are a group of aquatic microorganisms that have as a characteristic the magnetic nanocrystals that have a biological membrane called magnetosomes. They can be cubocathodic, elongated, prismatic, and bullet shape. Their size varies. The main advantages of applying this technology, I would like to highlight they are naturally biocompatible due to this biological membrane. They are produced in bioreactors. They can be functionalized easily and controlled by an external magnetic field. The magnetosomes are described in the scientific community in various areas and can be used in biotechnology. But today I'm going to highlight their participation in drug delivery and hypothermia for treatment of tumors. We establish large-scale production of magnetosomes with the cost of that varies between $17 and $170 per gram. Currently, synthetic magnetic nanoparticles need another stage which increases this price by 5,000%. So our magnetosomes are low-cost particles which makes them applied for the unified health system. Since 2017, the LabMax group has invested in magnetosomes as a new tool to improve the quality of health and human health in a variety of areas. And today I'm going to highlight how it could be used in neuromedicine. So how do magnetosomes contribute to neuromedicine? They can contribute to the treatment of neurodegenerative diseases like Alzheimer's and Parkinson's, which are common causes of morbidity and cognitive impairment in adults. And in Brazil, it is estimated that in 2050 there will be an increase of 500% in cases of Alzheimer. When we use the magnetosomes with the cortical neurons, we see they are non-pyrogenic, non-toxic, and stimulate the neurite outgrowth and increase synapses if they are internalized up until 24 hours. We can see this in this 3D reconstruction. So for the first time, we have stated that magnetosomes can be a tool in nanotechnology to treat diseases like Parkinson's or Alzheimer's. Magnetosomes can be a tool to treat glioblastoma, a brain tumor that has a difficult treatment. Magnetosomes can be formulated or not by drugs and controlled by magnetically in a specific region of the body. When associated with magnetic hyperthermia and an alternated magnetic field, these nanoparticles induce the local drug release onto the tumorous cell and can eliminate the tumor in up to 350 days. Currently, the Labomax group is conducting these clinical trials of the magnetosomes with people in the University Hospital of the Federal University of Rio. So based on the natural biocompatibility, low cost, high stability, the capability of magnetic properties, the magnetosomes that we are presenting here are a new tool in technology to expand these treatments in neuromedicine. I'd like to highlight that this is not just for brain tumors, but other kinds of tumors as well. Our team is in the Federal University of Rio microbiology area, coordinated by Professor Fernanda Abreu, collaborating with Luciana Romão and Flávio Garcia of the Research Center. So the magnetosomes are a novel nanotechnology tool that can be used in the unified health system and promotes the improved human life and health. Thank you. Thank you to all the participants for their presentations, and now we will be opening this up to questions, comments, of course, first of all from the examining board and then from our audience. So I'd like to pass the floor here. Who wants to start? Do you want to make a question? Okay, let's start with the examining board questions. Well, once again, congratulations to everyone. I don't know if those of you who presented had the opportunity of being here yesterday. I can't remember if I saw some of you here, but I thought the presentations were well structured. You were able to communicate your idea. And like I said yesterday, the challenging of exposing yourself is very big. It needs to be defeated in this startup process. And what you're doing here is very important. Regardless of the prizes, I think Fiocruz has the possibility of starting many partnerships, of using these ideas and taking them forward to society. So I think this is a great movement here. I have a doubt, and then I will make my observations after the other questions. My doubt is for Karina and Attila. Your business is based basically on philanthropy, but I couldn't see exactly the sustainability of this philanthropic process. Is that clear? I don't know if I got lost in some moment, but how do you want to work with the sustainability of this idea so that it can reach this philanthropic objective? As we said, with the team that we have, we can already take on 10 projects in the year. So our idea, like Attila said, for Venturini IP, we can have this international partnership, and it's very important for our business because since we don't have so many patent requests here in the country, we can't send a lot abroad. So it's important for us to start these partnerships. For Venturini, this would be the advantage. So even though this part of the process is, we're delivering our work, we will have a return with these international partners who will also see us as partners here. And that's the central part of this issue. And how would we be able to sustain this? Well, we can't increase this number. What we hope is to be able to maintain these 10 projects, because we need to continue working well. And if we were able to find some funding from abroad or some kind of investment, then we might be able to recruit more people to help us and be able to maintain this process. I think that's it. We need to identify, these are two separate issues. Venturini is a partner, even though it created iPush. iPush has philanthropic character, but it has this partnership with Venturini, who can donate these professionals for 10 projects annually. And in order to maintain this, or to make this grow in biotechnology, we would sustain this with external investments, hire other professionals to conduct these services and these patent services. I had a similar question which regards the technical viability, the financial viability, because even though this is a philanthropic operation, it's important to think about how this is feasible financially. And I have this question, and I think it's not just for you. I think it applies to all of the pitches. How is this economically feasible? Even though the objective might be philanthropic, the economic issue is still important here. We are committed to these 10 projects to keep this philanthropic project in a smaller level. If we could have more investments, we could have greater reach. But so if we had a bigger project with a higher dimension, we would need external investments. Just to add something, we have the structure, we have office spaces, we have the technical knowledge. André is the founder of our office. He has a lot of knowledge, lots of years of work experience. I started as his partner. I have 10 years in the biotech area. And what we're going to do to start really is donating our time and our knowledge, everything we know about patents, creating patent requests. It's not the same thing as writing an article, of course, so you need to know the area. We are going to donate our time for these initial stages. It's very important. We're going to donate even the deposit. And if we can make this process of totally philanthropic, then we would need investment. So I'm going to open this up to our audience, questions or comments, and then the examining board will make the final comments afterwards. Hi, good morning everyone. I'd like to ask a question about the technology of the magnetosomes. Congratulations, it's very interesting. I'm a microbiologist as well, and I have a doubt. Would you put this bacteria, if you were to put this in the brain, how would you do this? Because the passage is through the, would it be through an injection? Or is there a risk of getting meningitis? Because this is a bacteria that doesn't have an immunological system to defend itself. So how are you working this issue? We don't use the bacteria itself. We isolate the magnetic crystals and conduct this with the isolated magnetosomes. So this biological membrane is free of these toxins. We don't have this immune system issue. We work with stability tests of this nanoparticle and the quality of the material. These are resistant even to temperature variations. We have submitted them to 400 degrees Celsius and they are intact. So this is high quality material. Data from literature shows that they can be eliminated in urine, and in the UK and Mexico they have already showed that these nanoparticles are found in the human brain, even because of pollution or contamination. So the human being can live well with these nanoparticles, and the advantages of the magnetosomes is that they are stable and they don't need a repeated dose for the treatment. We think one dose would be enough to establish the treatment. Just one more question. Do you have the data about this, the lifespan of this in the brain tissue? Not yet. We are conducting clinical trials, and we are part of the nano health team of the state of Rio, along with the Brazilian research team, and we will conduct these studies with labs along with Fiocruz as well about the lifespan. We will conduct these studies and trials. Thank you very much. Can I ask one more question? Congratulations, congratulations to everyone. All of the pitches were very interesting, but I wanted to ask a question to Gabriele about the magnetosomes. How does this delivery work into the neural tissues? How does this transfer happen? Would it be possible to deviate this so it goes into other organs or tissues? And the other question is, can you encapsulate any kind of drug inside of this nanoparticle? Can it be used for biological drugs or just chemical drugs? Thank you for your question. So the magnetosomes can be functionalized in a variety of ways with molecules and drugs and antibodies. And the drug delivery occurs because when we use this magnetic field, the agitation occurs in this local nanoparticle, and the local temperature promotes the release of this drug into the location. In our local model of studies, we are testing this for breast cancer. It could be used for prostate cancer, a variety of tumors, and we are conducting trials for microbial activity and leishmaniasis and other diseases in cats that are very common in Rio de Janeiro. So there is a wide variety of possible uses, and not just a medical area, an industrial area. It can also be used in the bioremediation of residues of chemical industry. So working with these chemical compounds, we have this nanotechnology that is produced by a low-cost bacteria that is highly stable and can be applied to a variety of areas in health.
Bom dia, a minha pergunta é para Maria de Lourdes
My
sobre a
question is to
FC
Maria de
binding proteins.
Lourdes about the binding proteins.
Até aqui.
Okay, she's there.
É muito interessante, eu acho que proteína A e
It's
proteína
quite
G,
interesting.
para
I
quem trabalha
think
com anticorpos,
proteins
são as mais
A
usadas,
and
realmente
G
sempre
are
tem
the main
alternativas
used
de
if you
baixo
work
custo
with
interessante.
antibodies, and
E eu
we
tenho
have
duas perguntas.
low-cost
Uma,
alternatives.
quando você funde
But my
com estradiolina, para você
question
depois, na purificação, você
in
conseguir
the
pescar
purification, can you
o conjugado,
get
né?
the
Mas se
conjugate,
for
right?
para fazer
But
ELISA, por exemplo,
what
você poderia
about for
conjugar com
ELISA? Could you
HRP, com
conjugate
peroxidase pro
with
teste de ELISA?
HRP
E a
or
segunda pergunta é
peroxidases
se
for
vocês
the
pensam
ELISA
também,
test? Or the
além da
second
proteína A
question,
e G, existe a proteína L,
we
que se
still
liga
have
à cadeia
protein
porque eu
L
trabalho, por exemplo, com
linked to
anticorpos
the
não completos, que seria o
light
SCFG,
chain,
que é o
and I
VH e
work
VL.
with
Muitas vezes não tem porção
non-complete
FC.
antibodies,
A
and they
empresa
don't
pensa
have
em
the
também
FC
desenvolver produtos
portion.
não
So
seja
do
só
you
FC
think of
específicos, mas
developing
possa ser
products
também
not only
para a
FC
porção
specific,
FAB?
but also for FAB portions?
A primeira parte da tua pergunta,
Well,
a
in
quimérica, ela tem essa
fact, the
porção
chimeric
estreptabidina, e
proteins
com isso a
has
gente tem a
this
possibilidade de
streptabidine
conjugar com qualquer
portion, and
molécula
we can
biotinilada. Então, a gente
conjugate with
acopla
any
com HRP
biotinylate
biotinilada,
molecule. So
ela já tá
it
acoplada,
is
né? A proteína ligante de GG já está ligada
binded there. We have the
HRP, a
ligand
fosfatase alcalina,
linked to
a
phosphatase or
então você
fluorophorus,
consegue
and
conjugar
you
qualquer
can
coisa,
conjugate
inclusive
anything,
na construção
even in
das colunas
building the
cromatográficas, em cefarose,
chromatography
agarose, biotinilada
columns
também,
in
que você
cefarose,
imobiliza
agarose,
as
and
resinas,
biotin
certo?
that
Então, a
immobilize the
gente utiliza para ELISA, com
resin. So we
HRP,
use
aqueles dados
for
ali que eu mostrei
ELISA
são,
and data that I
acoplados
showed
com
are coupled
HRP.
to HRP.
Vocês também,
But in the
nós podemos
case
comprar de qualquer outra empresa o composto butinilado, ou você já faz
you
a
can
conjugação
buy
de
the
Já faz a conjugação.
butanylate compound for any other country, or you make the
partícula de ouro também
conjugation?
para
No,
ensaios
we
de fluxo
make,
lateral.
and also for
A
gold
tua segunda
particles.
pergunta era
And your
o
second
que nós
question,
temos hoje em
well, what we
desenvolvimento?
have
Nós temos
today under
essa
development, we
proteína
have this
Y1902 para detecção de IgG.
Y1902 protein for the detection of IgG, and we have another protein for IgY.
Não sei se vocês viram ali no slide.
I don't know if you
ele
saw
não
in the
detecta,
slide, but it doesn't
soro
detect
de ave, né? Então, nós
bird
estamos desenvolvendo
serum,
essa
so
que
we
tem
are
IgY,
developing
mas,
that.
variantes que não
And
se
we
ligam
have
por célula
variants not
a gente não tem nada no
binding
momento,
to the
mas a
FC
gente pensa em
portion. We
desenvolver no
have
futuro.
nothing
No momento, a
in
gente não tem
moment, but we think about the future.
Se hoje eu trabalho com um laboratório de pesquisa, sou da
I work with the research lab, and we'd like to know if you can buy the protein. No, it's not in the market. We are finishing the stability test. Probably at the end of this year, next year, it will be on the market, but we can get into contact, and I can send you a sample.
Não. Eu tinha uma pergunta para Maria, mas já foi respondida.
I had a question to Maria. It was answered. Congratulations. But to Andrea, I would like to know if in the marketplace you have services and technologies available other than high consumption material. Well, thank you for the question. The iBench market is the first solution of iBench, so we now have a lot of features added in the platform, not only lab products, but also services being contemplated. And then we are thinking about changing the iBenchmark in a big platform where scientists may find what they want in one place. May it be a robot or a platform to find the events that are happening and other features that we are implemented daily, but there is no date now. Do I have anybody else here on this side with questions?
Olá, bom dia, Maria de Lourdes.
Good morning, Maria de Lourdes from the protein binding to the antibody cells. I think most of what I would like to ask he mentioned, so I would just like to say that we are interested in being part and collaborating with you because as we have platforms to use monoclonal antibodies, purification is a very important step, and many times protein has the best solution. So in this sense, we are open to help and collaborate with you. I think I made this question again, but it will be very important for you to think about the scale process going from the benches, assays with 50 milliliters of supernatant to purify the antibody until the scale production for the industry where you have bioreactors of 50 to 100
Obrigada.
Thank you.
Esclarecendo a tua questão da escalabilidade, nós estamos desenvolvendo um projeto.
We are developing the project and we have a call for bidding for CNI in innovation, and we have collaborations of two CNI institutes in Rio, biosynthetics and green chemistry, and they are working in the production of the chromatography column and scalability. So at first we work with lab scale in small volumes, but scalability and production and all the tasks for larger scales will be carried out by our partners in the two ISIS in Rio. So we believe in the end of the project, which takes 2 years, we'll have the scalability ready for us to access the big scale market. Okay, thank you. Any other questions from the audience? No. So now I pass on the floor to the final considerations. I call the examining bench.
Pegando pouco o gancho das minhas considerações de ontem.
Well, taking my considerations of yesterday, today, I really could understand better the business plan that you are proposing. I understood better your goals, what you have with the space, and I could have more to evaluate that.
Falta de ter modelo de negócios.
I missed some business plans yesterday, a lot of scientific weight without thinking about what is exactly important, how the demand comes and how to solve the problem, and how is the structure of that, if I have a robust team and if it's viable. So today I could see this much better. So congratulations to you. I don't have anything else to say. We'll gather to evaluate you later on, but much more than the prizes, it's important to be present here and have your networking and collaborate in many other ways. And Fiocruz is always open to new ideas, new collaborations. And thank you all for your participation.
Boa tarde a todos. Eu queria agradecer o convite.
Good afternoon, everybody. I would like to thank for the invitation to be part of this evaluation, and it's quite interesting because we can see that currently you tend not to be limited to the research and labs and publications. I think expanding and making other researchers' life easier is very important. I was quite impressed with the diversity and the ideas that come and that can make your lives and other researchers' lives better. So I would like to congratulate you all on your work, and regardless of the prize, create knowledge worldwide and bring interactions is more important. And the prize is also very good.
Boa tarde a todos.
Good afternoon, everybody. I would just like to thank the participation of you all here. And regarding João's speech, I was yesterday here as well, and I could see that today we had more structured work. We know that it's different, it's quite a challenge to be here presenting the proposal, but I think that we had the opportunity to have this discussion in terms of technological gaps, and it's important to identify what would be our future partners and the ideas that we can make this space wider in next event. So thank you all for your presence.
Encerrando então mais uma sessão de pitch.
So we finished the last pitching session in this event. Thank you all for your participation. I thank the presenters for the presentations and also the examining board. And we see you in the next session. Thank you all. Congratulations to you.
Boa tarde, pessoal.
Good afternoon, everybody.
Estamos agora na última
We are
sessão
now
aqui do Innovation Hub.
in our last session in the Innovation Hub. We are in the parallel session that will be guided by Antero. You have his presentation, and we'll have the account manager of Clarivate, who will present the global alliances to solve personalized problems. So I pass on the floor to Antero for him to start the afternoon session.
Obrigado.
Bom, boa tarde a todas, boa tarde a todos.
Good afternoon, ladies and gentlemen. It's a pleasure to be here in the symposium. Congratulations to the organizer. The symposium is incredibly well organized. I see a lot of your participation. I know most of the audience is from Fiocruz. I know many of you. We've been together over the years, but for sure there are external researchers as well. So the idea is that we have new people here, people who doesn't know the company and what we do in terms of monitoring scientific and technological information. So I believe this presentation will add something to the reality that we have surrounding the symposium, and we'll try to have a brief overview of the pharmaceutical world and biopharmaceutical world and how we are moving regarding the partnerships that's being established and the main kinds of technology developed and how it is impacting the market and also impacting from the therapeutic point of view. And obviously in this general overview, you can imagine how important it is to understand everything for you to guide your research activities and institutional activities. Then we'll open for questions in the end. And please feel free to say anything. To start, I have a little bit of the scenario to present to you. I'll talk about biopharma in the current world, and some numbers are very important, and they are expressed here. When we think about what are the medications for one specific condition like infectious diseases that are under development today, we have innovative medications and we have more than 5,000 molecules between small compounds and biological therapies under development or recently launched with some innovative characteristics. And in this universe, we know that more than half comprise biological products in their different technological types of recombinant proteins and cell therapies and gene therapies and so on and on. We also know that we have lots of institutions behind the development programs, more than 3,000. And also thinking about the biologicals, we have an idea that at least 24 of them will overcome $500 million annual sales to 2024. So this is a brief overview of the efforts and some of the more important economic reflex that we'll have in the next few years. And I would say that almost all the development programs are not isolated in each one of the institutions. We are following the commercial curves established over the last years. And if we take a look at 2018, the numbers for this activity are quite huge. They decreased a little bit according to the total in 2017, but according to the economic volume, it surpassed. And these are some of the reflex of the commercial agreements. If we consider the main kinds of agreement, we can see that licensing activities are mandatory in terms of number, but they also reflect in about 26% in economic terms, and eventually for institutions like Fiocruz or academic institutions, in fact, in our country, it may be the more similar agreement to what we want. And it's followed by funding and acquisition activities that are more recurrent in the industry, among the main industries. Even in economic terms, we have more than 64% of all the economy of those $428 billion only last year. I mean, nothing moves alone. If we consider the therapeutical areas that are more searched and involved in this kind of agreement, we constantly have seen, and it is still true that we had oncology among the well-defined with the greatest activities in terms of commercial agreements. We see the amount of agreements regarding the partnerships in terms of merge and acquisition of companies,
followed by medications for neurological diseases, and then by agreements about infectious diseases. We saw a small growth between 2017 and 2018, but this is the panorama, and this is a very similar panorama to what we see relating to licensing. More than 40% of the agreements last year are in the oncology area, followed by neurology, infectious diseases, which we will talk about in a bit, and how these different therapeutic assets can serve not just to one, but more than one of these areas. So you can use oncology not just for cancer, but for infectious diseases as well. And one of the most important characteristics to consider regarding the agreements, if we look at the main agreements, they have a precocious phase in the development of the products. So if we look at the main licensing, 20 pieces of licensing last year, 13 had to do with products or prototypes in preclinical development stages. So very close to the reality that we have today in our lines of research in Brazil, or with our public at least. And in these agreements, the financial volumes are very high. We know that many of these agreements had more than $100 million in these licensing activities. So this is a possibility to consider when we have a good asset. We need to look at the price, find the best price for it. If we take a look at the oncology area, since this has the largest amount of agreements, we will understand that for oncology, the compounds that are being developed have been licensed in their beginning stages of development. We will then understand that the main licensing agreements involved medication that within an immuno-oncology classification, this is something that is recurring, it is a tendency, and it is something we will see in the therapeutic point of view very emphatically. And if we think about these boosting regions like China, for example, there's a big difference there in relation to the agreements that are practiced. And who is determining this activity in Asia is China, without a doubt. So some other characteristics, not only thinking exclusively of the agreements, but how this history of agreements in the last few years have led to innovative medications that have arrived at the market and have been approved. There was something very interesting last year if we consider medication that was approved by the FDA. Let's take a look at this number. We see this 59 new molecular entities being approved in 2018, and this is 3 times the quantity of new molecular entities approved in 2010. And here we can see that for most of these molecules, they were characterized and entered the revision system of the FDA, being considered orphan drugs. So these are medications for those who treat diseases, and previously these modalities, these therapeutic modalities did not exist. So there are some revision practices in the FDA that have existed since 2014. Some new options have been added. The surveillance agency here also works with these accelerated modality options for revisions, and this has been accelerating the rhythm of activity in the pharmaceutical industry. This also generates some interesting characteristics, because even though we have seen an increase in the medications approved, we know that they are getting to less patients because they are orphan drugs, which in many cases treat neglected diseases or diseases that are rare, that have a low populational rate. So these are some characteristics we have observed in the last few years. If we take a look since 2007 until 2017, these 10 years, we see that the number of molecular entities being approved by some regulatory agency in the world has grown in a consistent manner. So these numbers are compared to 2007, and this increase has to do with the volume of sales of these medications around the world. And on the other hand, during these years you had a decrease in the investment for the development of these compounds. But in general, we are seeing this investment returning compared to what we saw before 2007. So in the 80s and 90s, we saw this reducement in what was involved with development projects, and now this is being partially altered. Another characteristic is that if we consider the medication approved from 2010 to 2017, we see they are in this line, and they reach a smaller parcel of the population. So this question about the orphan drugs is important, rare diseases are important, and we will see there are other characteristics as well in these new medications. And they have to do with this decrease of the number of people that the drugs are reaching. This is definitely a tendency in the industry. It is intrinsically related to the processes of development of these medications and their clinical validation. Another important point is to start understanding what are the main players in this scenario. So we have the essential benchmark here to know how our country compares to others, how these entities participate in the development programs, and to understand what are the characteristics in these chemical compounds and the biological ones as well, because we understand there is a difference here.
Se analisando.
This analysis was done looking at the technological output for patents between 2015 and 2018. Here we see the list of the companies that created these patents for small compounds. and the number of patents conceded during this period. If we make a general consideration, we can see the greatest contribution comes from the pharmaceutical area, followed by 20% by non-commercial entities. So these are universities, research centers, public centers, funding agencies, foundations, etc. And when we compare this reality to the biologicals, we see a different scenario. Number one in the ranking is the University of California and a variety of universities. And when we analyze these numbers globally, we see that almost 60% of patents among the top 30 players in this segment come from non-commercial institutions. So for the biologicals, really what we're talking about gets close to these lines of research that we find in our scenario here in Brazil in public institutions. A few other comparisons that I think are valid for us to start understanding more about the characteristics of the biologicals facing the small compounds. It has to do with the mechanisms of action of these medications, which for sure in some scenarios are related between these two classes of medications, but there are specific characteristics for these groups. And this is very interesting if we consider the main targets, the main emerging targets, the pharmaceutical targets that had the biggest pharmaceutical involvement and development in the last years, the last 2 years compared to the last 10 years, we see BTK looking at small compounds as the biggest target, then BLD4, the general modulator of compacting activity and decompacting of chromosomes. Then we see other proteins as well, that in terms of epigenetics is one of the main players. So these are some of the main targets, emerging targets that we observe today, looking compared to the small compounds. When we look at the biologicals, the scenario is very different. Some proteins that are intrinsically related to immunology, like TGT, proteins like the envelope for Zika, are some of the main targets that we have tried to modulate these activities, especially through biological medications. So now that we understand there are some specific characteristics of the biologicals in relation to the action mechanism and looking at the diversity that exists in the technologies involved, and how these biological medications are handled, we have a huge combination of elements here. And it's interesting to see how these technologies are being ranked, which are being used the most, which are participating in the big development programs today. And first of all, we find the humanized monoclonal antibodies. They are at the top of this category, followed by human monoclonals. And then we have the vectors, the gene therapy vectors. This was born in the 80s. We had the first approvals in the past years, and we can see that these vectors, these virus vectors, are very high in this ranking. We have other technologies involving antibodies, biospecific antibodies, for example, conjugated compounds, antibodies that are here consistently, and also we still have the presence of cell therapy. We can still classify this according to the technology. What are the main mechanisms of action? The antibodies in a general way, considering cell therapy, a variety of cell therapies that are related to the modulation of the immune system, and genetic therapies. And we find some common elements between these different groups. We have some that are in the gene therapy and antibodies, but it's interesting to see that some of the main targets are related to the modulation of the immune system, and they are being used in immunology. So as I said, this is a tendency that whether you have a technological tool used to develop these medications, this is a line of work and of functioning that has been very efficient, and there is a great amount of research and resources being centered in this kind of idea. And I think we shouldn't just speak about the development of products. It's interesting to see how this development converges with what effectively will reach the market, or what is reaching the market. And every year Clarivate conducts a survey that lists the main blockbusters here of medications that will have a volume of more than $1 billion in the next few years per year, considering medication that will be launched in that specific year. If we look at the list of 2019, we see a list of 7 medications. There were 12 in last year's list, so this year we have less blockbusters, but usually these are biological medications, and the direct application of the technological advancements are here in the projection of the economic impact that they will have from this year on, with a sales projection up until 2023 between $1 billion to $2 billion. So if we could not prove this clinically, prove its efficiency clinically, then we would not see this scenario.
And we are not saying only the monoclonal, but also recombinant proteins, especially for even for allergies to peanuts, for example, going through cell therapies and reaching the therapiogenics, the gene therapies with this kind of vector that is very efficient for differentiated tissues. When we also look at the main diseases with the highest focus over the last years, we see the amount of diseases emerging with more medications being developed in the last years. So we see that among the diseases, some kinds of cancers are the most representative ones, like breast cancer, triple negative breast cancer, that the therapeutic practice is quite complicated, spleen cancer that with a high mortality, and so on and on. So it's interesting to know a little bit of not only the technology and how it's developed, but also how to work with what kind of disease, because things go in parallel. Fortunately, not only in terms of technological analysis, we see more and more the diversity and new targets being approached, and also the clinical reflexes of this kind of technology that are more and more effective.
Esse aqui é levantamento bem interessante.
This is a very interesting research, as we are talking about cancer that much, and this considers the therapeutic management of different tumors in a period between 89 and 93 in green, and in period between 2018 and 12 in blue.
tem se mostrado de forma cada vez mais
how it
efetiva.
has
Então,
been
essas barrinhas
more
aqui
and more
têm a ver com o
effective.
percentual
So
de
the bars
pacientes
here
livres da
has
doença
to do with
depois do seu
the
diagnóstico no
percentage of
período de 5
patients
anos.
free from cancer after the
E a gente
5-year
consegue ver aqui
diagnosis.
para vários tipos de
And
câncer,
we
de
can
forma
see
geral,
that
existe
for many types of
digamos,
cancers in
uma
general,
sobrevida,
we
a gente
have
poderia, colocar
a
dessa forma,
survival
maior,
that we
e
can
pra
say
grande
is
parte
higher,
desses
and for
tumores,
most
de
of the
forma mais
tumors
isolada,
in the
isso também
more
é verdade.
isolated
Ou seja,
way,
os
it's
ganhos
also
terapêuticos
true. So the
estão
therapeutical gains
têm acompanhado os ganhos
have followed the
tecnológicos,
technological
né?
gains.
Isso é meio óbvio a gente
And
pensar,
it's
mas
quite
a gente, quando
obvious
fica
for
na
us
dúvida
to
de
think
até
about
onde
it,
vai o
but
interesse
as
da
we
companhia, da
are
indústria
in doubt
farmacêutica,
about the
onde
interest of the
começa a real
pharmaceutical companies
aplicabilidade
and the
e ganho
real
clínico, acho
applicability
que
and
esse
clinical
infográfico
gain.
diz
So in this
bastante,
sense, I
né?
think this graph shows a lot.
E eu não comentei no início, mas a gente
I
não tem
didn't
nenhum tipo de
mention
conflito de
before,
interesse
but we
com a
don't
indústria,
have
então
any
me
kind
sinto
of
livre,
conflict
né,
of
para comentar o
interest
caso
with
de
the industry,
desses
so I may
biológicos
comment the
mais
case
recentes
of
que
one of the most
tem se
recent
demonstrado muito
biologicals
eficiente para
that
alguns tipos
has
tumorais,
proven
que
quite
é o
efficient for
Keytruda,
some
né, que é
tumors, that is
anti-PD1, por
Keytruda, that
exemplo,
is an
algumas das suas
anti-PD1,
características
and one of the main
refletem
characteristics
muito,
reflect
de como outros biológicos
how other
têm se comportado hoje.
biologicals
Então,
are
a
behaving
gente vê que
nowadays.
realmente
So we see
muitas
that
dessas
in fact
moléculas
many
geram
of these molecules
ganho
generate a
clínico
clinical
muito
gain
superior
that
em
is
relação
quite
ao
higher
tratamento
regarding
padrão,
the
tratamento
standard
convencional,
treatment, the
né?
conventional one.
Quando administrado,
When
seja de forma isolada, quando
used
em
in isolated way
eventualmente
or
até
in
com agentes
combinations, even with the chemical
E quando a gente
agents.
começa a olhar pouco mais
And
das
when we look a little
especificidades
bit
em
more
relação
about the
aos pacientes,
specificities to
a
the
gente encontra
patients,
cada vez
we
mais
see
afunilamento
more
maior.
and more
Então, a gente viu
bottleneck.
no
So
passado,
we
né, em
saw
alguns
in
slides
the
atrás,
past
que a quantidade de
that
pacientes
the
sendo
amount of
atendida,
patients
né,
treated
estava diminuindo nos
was
últimos
decreasing
anos.
over
A
the
gente
years.
justificou isso em
We
parte
justified
pelos
partially
nichos de
by the
doenças,
niches of
raras que
rare
começaram a ser
diseases
mais
that
atendidos,
started being
mas também
treated
outro
as
fator
well. But
super
another
importante para
important
esses
factor
tipos
for
de
this
medicamentos
kind
tem a
of
ver com
medication
a
has to
população
do
ou a
with the
subpopulação
population or
que está sendo
subpopulation
atendida por eles
being
dentro
treated
de
by them
populacional que
in this
apresenta
population
o
group
mesmo tipo de
presenting
doença,
the same kind
pelo
of
menos
disease.
do ponto de vista
At least from the
histopatológico, né?
histopathological
A gente
point
vê
of
que, por
view, we
exemplo,
see that,
as
for example,
prescrições para
the
Keytruda
prescription for
exigem que os
Keytruda
pacientes apresentem
demand patients to
alto nível de
present a
PDL1.
high level
Isso
of
tem a ver diretamente com o
PD-L1.
seu
It
mecanismo
has
de
to
ação,
do
né?
directly with the
E que, ao mesmo tempo,
mechanism of
sejam
action,
pacientes
and
que
at
não
the same
apresentem
time,
mutações
patients
em
with no
EGFR
mutations
ou
in
ALK.
EGFR or ALK,
Provavelmente,
because probably
são efetores mais downstream da via
these
de
are
transdução,
more
e se
downstream
tiverem mutados
factors,
constantemente
and if they are
ativos, não adianta você
constantly
bloquear,
mutated and
lá em cima,
active,
porque
you
a
cannot
transdução
block
vai
up there
continuar,
because the transition
muito ativa,
will
né?
be
Isso
really
a gente
active,
aprendeu já
and we learned
com
that
o
with
gefitinib, a RISA, né, o
gefitinib
composto
or
químico lançado, que foi o primeiro
NISA,
inibidor de
which was
EGFR
the first
há muito tempo, que
PDFL
no
inhibitor that
pós-lançamento se mostrou
after
muito
being launched
inefetivo,
was quite
principalmente em relação a
ineffective,
isso, né?
especially
Eles não
regarding
estavam
that,
conseguindo
because they
distinguir
couldn't
o grupo
distinguish
populacional,
the
daqueles
populational
pacientes
group of patients
de tumor de pulmão
with
que
a
poderiam se
lung
beneficiar
tumor that
mais
could
em relação
benefit
ao uso
more
do medicamento.
regarding the
Então,
use
essa
of
aqui
medication. So
é a nossa medicina
this
personalizada,
is our
né? Ou seja, é braço
customized
daquilo que
medicine.
tem
This
sido explorado
is
como
one
medicina
branch of
personalizada,
what
como
is
que
being
a análise do perfil
explored as the
molecular
customized
de cada dos
medicine and the
pacientes, mesmo
analysis of the
apresentando,
molecular
o mesmo
profile
tipo de
of each
doença
one of the
classificada,
patients, even if they
pode
have the
e
same
deve ser
classified
usado
disease,
pro manejo
and
terapêutico.
how it can be used for the therapeutical management.
Então esse infográfico
So
reflete
this
pouco disso,
infograph
né? Como
reflects
que,
a little bit of
eventualmente,
it,
a gente conhecendo as
how
características
eventually we
dos
know the
pacientes,
characteristics of patients,
tanto do ponto de vista
both from
genotípico,
the
quanto,
genotype point of
na busca de
view
outros tipos de
and in
marcadores,
search of
pode
other
facilitar
markers,
no
and
sentido
how
de se
it can
encontrar
facilitate
populações ou subpopulações que
finding
respondam cada vez
populations
melhor
or
e
subpopulations
apresentando menos
responding better
toxicidade
and
à
better
coleção
and
de
with less
medicamentos.
toxicity
Inclusive,
to a
muitos
collection of
estudos hoje na área
medications,
de
even
câncer, pelo
many
menos os
students in
chamados
cancer today,
basket
or at least the
studies,
ones
né,
called
estudos
basket
clínicos,
studies,
têm
the clinical
consideram isso como
trials,
ponto
they
de partida,
consider
ou
that
seja,
the
agrupar os
starting
pacientes de
point, I
acordo com
mean, in grouping patients according to the molecular profile and not necessarily with the kind of disease or the kind of cancer they are presenting. And with this molecular profile, finding or proving the efficacy and the efficiency of the medication.
E dentro de tudo isso que a gente comentou,
And regarding everything that we mentioned here, I think that for those of you who work in the field, you know, but more and more it's worth mentioning that all or at least most of the programs of pharmacological development have the target as the starting point. It's important not only to establish effectively targets with a cause correlation that is quite direct to the kind of disease. This is key to understand the biology of the disease from the molecular point of view and know the main targets that, when emulated, may interfere in the phenotype, but also take into consideration what is the level of competition from the point of view of the target. How we deal in this scenario of development that we just commented, how can we look at the medications under development regarding their targets? What targets are more or less explored? What are the preclinical results and eventually the clinical ones of the targets being evaluated, and how to position regarding the development of a new therapy in this competitive scenario? We want to take more risk to develop medication for a well-established target but clinically proven as effective, or do we want to take more risks and explore not well-characterized targets with a chance of being innovative in this sense? And if we take a look at the amount of targets being explored now from the point of view of drugs under active development, it's quite expressive. I would say that 1/10 of the human proteins that we know so far. Another important characteristic is regarding the development of clinical trials in terms of grouping more and more patients. So if we have a comparison between the clinical trials and the amount of criteria of eligibility from 2000-2003 compared to 2008-12, we see that there was an increase of almost 50% in the eligibility criteria so that patients could be part of the clinical trial. I mean, from the point of view of selection, there is a higher pressure for smaller subgroups of patients. Added to that, we know that the industry in many cases has a follow-up of the genetic profile and other markers of the condition during the clinical trial, and during this period we can identify markers to help them classify the best or worst respondents. When we launch the medication, we have that facility of reaching something more specific with a more specific efficient response for a smaller group of patients. And if we take a look at the main markers approved or under development or in validation that may be used for the therapeutical selection, we see that just a few were approved, but we have a lot of research in this area.
Então, realmente, cada vez mais, não só marcadores,
not only markers reflecting more and more in an early way the diseases, but also markers to be used for the selection, the trial, the screening of patients. This is something that is being dealt a lot. So I hope so far I could have brought something to instigate your curiosity, and how can we eventually be in basic research and find information in clinical trials or financial information, for example, giving more basis to your research with the resources that you have, with what you can do based on this information, even if in basic research you can show thoughts that are more used from technological point of view. There are manufacturers, we mentioned some, but there are much more. Understanding diseases from the molecular point of view is key. Also knowing which one of the proteins or molecules are used as biomarkers, and know if the biomarker is being approached from the therapeutical point of view as a target, knowing the medications under development that can be competitors or an opportunity for projects like reposition of medications, because we know the mechanisms of action and diseases that these medications treat, and why not eventually propose repositioning modalities, and to all that know the clinical trials in terms of efficacy and safety. There is all the analysis of safety and security of medications regarding the mechanism of action that is important if you consider as the study, consider everything from the starting point and the genetic characteristics of the population treated. This is what we do, this integral analysis, integrated analysis of the variants to supply the researcher and even people from new business in Fiocruz information from or people from the industry information to make their decisions, especially because they are not the only ones. We have a high number of other factors that may guide all this process. The regulatory issues that may vary from region to region, pharmacological or technological and diagnosis point of view, the commercial agreements, as we saw, that today are very important in the study. Patent analysis, we know that technologies are not covered only by information that we have in scientific literature, but emphatically also in patents and also projections of economic reality. This is what we try to do, and how Clarivate does that is structuring information. So we have printing groups and editorial groups that, from the point of view of the scientific literature, we have the open door, the entry door for the web science, and from the point of view of patents, as the entrance we have Delta Maior as the biggest patent group in the world, and in terms of regulatory analysis and conferences and so on and on, we have our editors being able to structure the information to bring to you adequate solutions to each one of the stages of the life of development of a product, from discovery to protection and commercialization. And just to finish, we have a short video that I think in 2 minutes will show you a little bit of what I'm trying to explain here for more than half an hour.
This is the pill that saved my life. Before that, it was manufactured by a pharmaceutical company. And before that, it was discovered by a scientist. What happened in between? Before it was a pill,
it was an idea. While on a research trip to the Amazon, I collected the saliva of a frog species. I discovered that it contained a key protein, and I thought that protein, if synthesized, might someday save millions of lives. Using Clarivate analytics tools, I researched the protein, wrote a paper, and presented it at a symposium
where I was in the audience. We connected and agreed that the idea could make a real difference. So I did a genetic pathway analysis to check the potential of the idea. And I asked product development to find out if another company
was working on the idea. I investigated and found out that there was nothing in the drug pipeline. At least not yet. So I contracted with Clarivate Analytics for a white space research report, which showed me that the idea would have a market. So we developed the idea into a drug, a pill, in fact, and I looked into the regulatory requirements before we tested it successfully. At the same time, we needed a patent.
I conducted global patent research online to help me write the patent application, and my team did trademark research to establish the uniqueness of the pill's name. It became a brand and we protected it. We marketed and sold the pill around the world and delivered it
to me, and it did. It saved my life.
contextualizar bem, né?
So I think in 2 minutes it's pretty easy to contextualize everything. I hope you enjoyed my lecture. I will be around in the afternoon. If you have any doubts, I don't know if we have time for questions and answers now, but we can keep talking after this. Thank you very much. Have you opened this up to questions and answers? We've reached our time now. We have 5 minutes for questions. We have to make this fast. Does anyone have a question or a comment or an observation? Everyone's quiet here. Thank you for your excellent lecture. First, I have a silly doubt. This data from the report, is this report available or is it paid? I need the speaker to use a microphone so I can make the translation. Sorry.
Tá. E aí tem uma...
I have a doubt. What we're doing actually, substratifying populations. So I had a big population that said they had lung cancer, and then I subtyped this and subtyped it again. And I have medications that work for group X but not group Y or Z, etc. So do you think that we will create new rare diseases, or do you think the big pharmaceuticals will cover this piece by piece and put this puzzle together? I think that question has a lot to do with, not just in this sense, but regarding the costs, the viability of everything that we are seeing start regarding the axis of the population. This is something we are already observing. Big pharmaceuticals are trying to cover these gaps. It's not a problem for them to treat less patients. They're going to look at the price, right? It's not a problem regarding the amount of doses. If we look at the treatment of HCV, for example, which was launched in 2013, 2014, one dose, the rate of intervention was almost 100%. And the biological that was used previously covered 50% in terms of response, and it was a lot of doses. So it's not a dosage issue or a population issue, but there's always a price issue here. I think what we need to look at is how the population will be able to receive this care. Let's look at health care, the ethic demands in the future, if you will have health insurance or not, public health, like here we have the unified health system, how accessible these more expensive or more niche medications will be. So we are stratifying all of this, and we will have to consider all of these elements. Just 2 more questions because they will have a meeting here afterwards. I have a doubt about in what moment Clarivate enters. Do you enter in the conception of the idea, or does the researcher already have an idea and then get in touch with you, and then you help to mold the whole system, or do you already have, or do you work with a product that is already started? Because I felt like you participated in many phases. So what kind of partnerships do you make with researchers? Is the researcher involved? Are they on the sidelines? So the first point is that we provide information. We don't have pharmaceuticals or anything like that. We provide information. And this information will be used by the researchers always so that they can make the decisions. So even in consulting areas that we practice, the decision always will end up being made by the professional, the researcher. So we have two big support modalities. One is with analytic tools like Web of Science, a general search engine, and then we have more specific tools for life sciences. So I'd say this is the main modality in our territory today. CAPES finances through this portal a super tool which covers more than 500,000 compounds in development and the area of markers and therapeutic selection markers. So for those of you who are in the Federal University of Rio, get in touch with me so you can take a look at this content. This is a huge investment by CAPES that can be very well used and can be more and more used. And then we have the consulting area that is independent. Well, whether it's consulting or the tool, I think the big issue is that even before you start a project, you have a profile of what's being developed in the world, and this is crucial. So for a pre-stage, before the development and during the development, you need to have the necessary benchmarks to finish the project, even to have make the literature clear. The tools are very interesting. They're on CAPES. You can use them. We have a partnership with Fiocruz that extends to other tools, to the area of commerce and clinical trials. But we have a whole series of other solutions that can also help, for example, systems of biology or compounds, and these are more analytic tools for research. Antero, good afternoon. Congratulations on the presentation. Thank you very much. I am from Biomanguinhos, and I wanted to ask you, these databases that you looked at, do they consider diseases by infectious agents, or were they concentrated especially on cancer and autoimmune diseases? Well, actually, maybe that was my mistake. I know I mixed a lot of subjects together. For some of these slides that I showed, we definitely covered the medications. They are separate from their therapeutic areas, and then for other areas, of course, we had a greater focus like oncology. So it depends on which slide you're referring to, but our analysis is complete. Covers everything, the most prevalent diseases to the rarest diseases. Yes, because it caught my attention, actually. I was worried to see there are no new molecules to treat infections by bacteria, especially these resistant bacteria that are killing a lot, and they say that by 2050 this will kill as much or more than what cancer kills today. So I was worried to see there were no promising molecules that are being developed to treat these resistant bacteria. I think this has a lot to do with the methodology that was used to make these analyses, and of course this is an area of research that, well, the projections are still being created regarding the economic impact. They don't seem to be as high as those in the oncological areas. Maybe that's why, depending on the analysis that was made, they were not considered. But yes, we are monitoring these resistant bacteria, and because you are from Biomanguinhos, of course, let's keep in touch and see how the tools can help these issues. I wanted to thank you for your presentation, Antero. Thank you for your presence. This is our last session at the Innovation Hub. Once again, thank you very much. We will have a session now at the auditorium. It is the session before the closing session, so we will have a lecture and then our closing lecture. Thank you very much. Have a good afternoon.
Vou chamar para ir lá ao fundão então para dar uma palestra.
Tem um